Have you ever wondered what secrets lie hidden within the rocks beneath our feet? Tonight, we're embarking on a journey through time that will take us deeper into the ancient world than most people ever imagined possible. While you settle in for a peaceful night's rest, let me guide you through one of the most remarkable chapters in paleontological history. The Earth holds countless mysteries, and sometimes the most extraordinary discoveries come from the most unexpected places. Picture this. Scattered across the globe, buried in sedimentary layers that formed millions of years ago, are fossils so perfectly preserved that they still contain the actual organs of creatures that lived when our planet was an entirely different world. These aren't just impressions or mineral replacements of bones and teeth. We're talking about the actual soft tissues, the delicate internal structures that once kept these ancient animals alive. Most fossil discoveries consist of hard materials that resist decay over geological time. Bones, teeth, shells, and other mineralized parts of organisms have the best chance of surviving the fossilization process. But every once in a while, under very specific conditions, something truly magical happens. The soft tissues that normally decompose within days or weeks after death become preserved in exquisite detail, creating windows into the past that reveal not just what these animals looked like, but how they lived, what they ate, and even how their bodies functioned. Tonight's journey will take us to three extraordinary locations where such miraculous preservation occurred. We'll explore fossils that have retained their internal organs for over 100 million years, specimens so well preserved that modern scientists can still observe the texture and structure of ancient digestive systems, respiratory passages, and even brain tissue. These discoveries have revolutionized our understanding of prehistoric life and continue to astound researchers around the world. As we begin this gentle exploration, imagine the vast expanse of geological time stretching out before us. The creatures we'll encounter tonight lived in a world where flowering plants were just beginning to evolve, where massive sea reptiles ruled the oceans, and where the ancestors of modern birds were still developing their first feathers. It was a time when the continents were positioned differently, when the climate was warmer and more humid than today, and when life was experimenting with forms and functions that would eventually lead to the incredible diversity we see in the modern world. The preservation of soft tissues and fossils is an extraordinarily rare event. It requires a perfect storm of environmental conditions that must align precisely at the moment of death and continue throughout the fossilization process. The animal must be buried rapidly, protecting it from scavengers and the usual processes of decay. The chemical environment must be just right, often involving specific mineral compositions in the surrounding sediment. Oxygen levels must be low enough to prevent normal decomposition, yet the conditions must allow for the gradual replacement of organic molecules with stable minerals. When these conditions do align, the results are nothing short of miraculous. Instead of just bones and teeth, we find fossils that preserve the three-dimensional structure of organs, the branching patterns of blood vessels, and even the microscopic details of muscle fibers. These specimens provide insights that would be impossible to obtain from traditional skeletal remains alone. Our first stop tonight takes us to the sun-drenched hills of Italy, where limestone quarries have yielded some of the most remarkable fossils ever discovered. In these ancient rocks, formed when the region was covered by warm, shallow seas, lies a treasure trove of perfectly preserved specimens. The geological formations here date back to the Cretaceous period, a time when the Earth was experiencing one of its warmest climates in history. The quarries northeast of Naples have been worked for generations, providing building stone for local construction projects. But it was an amateur fossil collector working in these humble surroundings who made one of the most significant paleontological discoveries of the 20th century. Little did he know that the specimen he carefully extracted from the limestone would revolutionize our understanding of dinosaur anatomy and physiology. The fossil in question belonged to a small theropod dinosaur, a member of the group that includes some of the most famous predators in Earth's history. But this particular specimen was different from anything that had been found before. It was a juvenile, incredibly small and delicate, measuring less than an inch in its preserved state. What made it extraordinary wasn't its size, but what it contained within its tiny ribcage. When paleontologists first examined this fossil under magnification, they could hardly believe what they were seeing. The preservation was so complete that they could identify not just the skeleton, but the actual internal organs of the animal. The heart, stomach, intestines, and even the trachea were still present, mineralized, but retaining their original three-dimensional structure. It was as if someone had taken a modern animal and turned it to stone, preserving every detail of its internal anatomy. The process that created this remarkable preservation is called phosphatization, a type of fossilization that occurs when calcium phosphate minerals gradually replace the original organic tissues. This process can capture incredible detail, down to the cellular level, but it requires very specific conditions to occur. The animal must be buried rapidly in sediment with the right chemical composition, and the environment must remain stable throughout the long process of mineralization. As we examine this ancient creature more closely, we can see features that would normally be lost to time. At the base of the neck, tiny C-shaped structures are visible, embedded within mineralized connective tissue. These are the fossilized remains of cartilaginous rings that once supported the animal's windpipe, allowing it to breathe efficiently as it hunted in the ancient coastal environments of what is now Italy. But perhaps the most striking feature of this fossil is the preservation of its digestive system. The intestines are visible in three-dimensional detail, described by researchers as having a lumpy, shiny texture that closely resembles what you might see when examining a modern animal. The level of detail is so extraordinary that scientists can distinguish between different sections of the intestinal tract, identifying the duodenum, jejunum, and ileum with the same precision used in modern anatomical studies. Following the path of the digestive system, researchers have even identified the rectum and, remarkably, an intact fecal pellet. This fossilized waste material provides direct evidence of the animal's last meals, allowing scientists to reconstruct not just what it ate, but the order in which it consumed its prey. The contents reveal a diverse diet that included both fish and small reptiles, suggesting that even as a juvenile, this predator was already an accomplished hunter. How? The preservation extends beyond the digestive system to include traces of the circulatory system. Faint, branching patterns visible on the surfaces of the organs represent the fossilized remains of blood vessels, the network that once carried oxygen and nutrients throughout the animal's body. This level of preservation is almost unprecedented in the fossil record, providing insights into the physiology of these ancient creatures that would have been impossible to obtain from bones alone. Near the front of the body cavity, researchers have identified a reddish stain that appears to be the mineralized remains of the liver. Chemical analysis of this region reveals high concentrations of iron-rich minerals, likely derived from the breakdown of hemoglobin in the animal's blood. This means that we may actually be looking at the chemical signature of blood that flowed through this creature's veins more than 100 million years ago. The fossil also preserves patches of muscle tissue in remarkable detail. Under electron microscopy, individual muscle fibers are visible, some showing the characteristic banding patterns that allow muscles to contract. These preserved structures provide direct evidence of how these ancient predators moved and functioned, offering insights into their strength, agility, and hunting capabilities. Even the animal's claws retain their original keratinous sheaths, the horny coverings that would have extended their length and sharpness in life. This detail reminds us that the weapons of these ancient predators were often more formidable than their fossilized bones alone might suggest. The tiny size of this specimen raises intriguing questions about its behavior and upbringing. At only a few weeks old, it seems unlikely that such a young animal could have successfully hunted the fish and reptiles found in its stomach contents. This has led some researchers to suggest that the fossil may preserve evidence of parental care, with adult animals providing food for their offspring, much as modern birds and crocodiles do today. As we continue our journey through these ancient preservations, we travel eastward to the fossil-rich deposits of northern China, where another extraordinary discovery awaits. Here, in sedimentary rocks that formed during the early Cretaceous period, paleontologists have uncovered specimens that rival even the Italian discovery in their remarkable preservation. The Chinese fossil beds are famous for their exceptional preservation of feathered dinosaurs, specimens that have revolutionized our understanding of the evolutionary transition from dinosaurs to birds. But among these remarkable finds is a specimen that preserves not just external features, but internal anatomy as well. This fossil belongs to a dromaeosaur, one of the raptor dinosaurs that has captured the public imagination through popular culture. The specimen measures about 5 feet in length and was likely covered in a thick coat of feathers during life. Impressions of this plumage are still visible along the head, neck, back, and tail, adding to the already extraordinary nature of this find. But it's what lies within the body cavity that truly sets this fossil apart. A distinctive bluish layer is visible in the lower abdominal region, extending forward through the body in a pattern that closely resembles the intestinal preservation seen in the Italian specimen. When examined under high magnification, this layer reveals itself to be composed of densely packed microcrystals formed through the same phosphatization process that preserved the Italian dinosaur's organs. The similarity between those two fossils, separated by millions of years and thousands of miles, suggests that the digestive anatomy of carnivorous dinosaurs may have been more conservative than previously thought. Despite belonging to different lineages of theropod dinosaurs, both specimens show remarkably similar arrangements of their internal organs, indicating that successful predatory strategies may have evolved early and remained largely unchanged across different groups. Interestingly, neither fossil preserves any trace of the stomach, suggesting that the highly acidic environment of this organ continued to break down tissue even after death. This selective preservation provides insights into the different rates of decay affecting various organs, helping scientists understand the complex processes involved in exceptional fossilization. The preservation of internal organs in these fossils represents more than just anatomical curiosities. They provide direct evidence of how these ancient animals lived, moved, and interacted with their environment. The structure of their digestive systems tells us about their dietary preferences and hunting strategies. The preservation of muscle tissue reveals details about their strength and agility. Even the chemical signatures left by their blood provide clues about their metabolism and physiology. As we move toward our final destination tonight, we shift our focus from the organs of the body to the most complex and mysterious organ of all, the brain. The preservation of neural tissue in fossils is extraordinarily rare, as brain matter is among the most fragile and rapidly decomposing tissues in any animal's body. Yet, occasionally, under the most exceptional circumstances, even this delicate tissue can be preserved for millions of years. Our journey now takes us to the windswept beaches of southern England, where the relentless action of waves and tides has exposed geological treasures that span millions of years. Along these coastal cliffs, where the sea meets ancient sedimentary layers, amateur fossil hunters and professional paleontologists alike have made discoveries that continue to reshape our understanding of prehistoric life. It was on one such beach, beneath the gray skies of the English coast, that a discovery was made that would challenge everything scientists thought they knew about the preservation of the most delicate biological tissues. The fossil appeared unremarkable at first glance, resembling nothing more than an ordinary brown pebble worn smooth by countless years of wave action. Yet this humble specimen would prove to contain something that had never been found before in any dinosaur fossil. The rocks along this stretch of coastline date back approximately 133 million years, to a time when Britain was positioned much closer to the equator and enjoyed a warm subtropical climate. During this period, the region was home to large herbivorous dinosaurs that roamed through lush forests and along the shores of ancient seas. These were the iguanodonts, robust plant eaters that could grow to impressive sizes and possessed the distinctive thumb spikes that made them formidable despite their peaceful nature. When researchers first examined this particular fossil under magnification, they realized they were looking at something unprecedented in the annals of paleontology. What they had discovered was not just another endocast, the typical stone mold that forms when sediment fills the empty space left by a decomposed brain. Instead, they were looking at actual preserved brain tissue, the mineralized remains of neural structures that had somehow survived the passage of geological time. The preservation of brain tissue represents perhaps the ultimate challenge in fossilization. Unlike bones, teeth, or even organs like the heart and liver, brain matter lacks any hard structures that might resist decay. It consists primarily of soft, fatty tissue that begins to decompose within hours of death under normal conditions. For such delicate material to survive long enough to become fossilized requires a perfect alignment of environmental factors that occurs perhaps once in tens of millions of fossil specimens. The process that created this remarkable preservation likely began when the dinosaur died near a shallow, stagnant body of water. The specific chemical conditions of this environment, probably acidic and low in oxygen, created a natural preservation medium that prevented the normal processes of bacterial decomposition. The animal's head became buried in fine sediment at the bottom of this ancient pond or lagoon, where the unique water chemistry began the slow process of replacing organic molecules with stable minerals. Under the electron microscope, the fossil reveals structures that would normally be invisible to the naked eye. The outer membranes of the brain, known as the meninges, are preserved in remarkable detail. These protective layers, which in life would have cushioned the brain and provided it with nutrients, have been replaced molecule by molecule with calcium phosphate minerals. The same phosphatization process that preserved the Italian dinosaur's organs has here captured the most delicate neural tissues with extraordinary fidelity. Within these preserved membranes, researchers have identified tiny mineralized fibers arranged in ribbon-like patterns. These structures represent fossilized collagen bundles, the protein framework that once formed the protective outer layers of the brain. In some areas of the fossil, different textures are visible, resembling the arachnoid matter, another protective membrane that in modern reptiles lies directly against the brain surface. Perhaps most remarkably, within the preserved membrane structures are faint impressions that appear to represent the actual surface of the brain itself. The ridges and folds characteristic of neural tissue are visible in microscopic detail, suggesting that this fossil preserves not just the brain's protective coverings, but fragments of the gray matter that once processed information and controlled behavior in this ancient herbivore. The implications of this discovery extend far beyond the simple fact of preservation. In modern reptiles, the brain typically occupies only about half of the available space within the skull, with the remainder filled by blood vessels and air-filled sinuses. Because of this, scientists have long assumed that dinosaur brain endocasts primarily represent the empty space around the brain rather than the brain tissue itself. However, this particular fossil suggests that the brain matter pressed closely against the inner surface of the skull, with less than a millimeter of space between the neural tissue and the bone. This observation raises intriguing questions about dinosaur intelligence and brain size. If dinosaur brains filled their skull cavities more completely than previously assumed, it would suggest that these ancient animals may have possessed larger, more complex brains than scientists have traditionally believed. The cognitive capabilities of dinosaurs have been a subject of ongoing debate, with recent discoveries suggesting that many species exhibited sophisticated behaviors, including complex social interactions, elaborate nesting behaviors, and even tool use. However, researchers caution that this apparent close fit between brain and skull might be an artifact of the fossilization process itself. As the brain tissue decayed, it may have collapsed inward, pressing against the protective membranes and creating the illusion of a larger brain than actually existed. Without clear preservation of the brain's internal structure, it remains impossible to determine with certainty the true size and complexity of this ancient animal's neural capabilities. The chemical analysis of this fossil has revealed additional fascinating details about the preservation process. The mineralization appears to have occurred in stages, with different tissues being replaced at different rates. The outermost membranes were the first to be mineralized, followed by deeper structures as the fossilization process progressed inward. This sequential preservation has created a layered record of the brain structure, with each layer providing different types of information about the original anatomy. The discovery of fossilized brain tissue also provides insights into the evolutionary relationships between dinosaurs and modern animals. The structure of the preserved membranes suggests that iguanodonts had a brain organization more similar to modern crocodiles than to mammals. This finding supports the close evolutionary relationship between dinosaurs and their modern archosaur relatives, while highlighting the differences between dinosaur neuroanatomy and that of mammals. As we examine this extraordinary fossil in the quiet hours of the night, we can appreciate the remarkable chain of events that led to its preservation. The dinosaur's death near that ancient pond, the specific chemistry of the water, the rapid burial in fine sediment, and the millions of years of geological processes that followed all had to align perfectly to create this unique window into the past. The fossil represents not just a scientific curiosity, but a direct connection to the thoughts and experiences of an individual animal that lived in a world vastly different from our own. The study of this specimen continues to yield new insights as analytical techniques improve and our understanding of fossilization processes deepens. Each examination reveals additional details about the structure and organization of dinosaur brains, contributing to our growing knowledge of how these ancient animals perceived their world and interacted with their environment. The rarity of such preservation makes each discovery all the more precious. While thousands of dinosaur fossils have been found over the past 2 centuries, only a handful preserve soft tissues, and even fewer retain traces of neural structures. This scarcity means that each specimen must be studied with the utmost care and precision, using the most advanced analytical techniques available to extract every possible piece of information. The techniques used to study these fossils have evolved dramatically over the past few decades. High-resolution CT scanning allows researchers to examine internal structures without damaging the specimen. Electron microscopy reveals details at the cellular level. Chemical analysis can identify the mineral composition and even detect traces of original organic molecules. These tools have transformed our ability to study ancient life, revealing details that would have been impossible to observe just a generation ago. The preservation of soft tissues and fossils also provides unique opportunities to study the evolution of complex biological systems. By comparing the anatomy of ancient animals with their modern relatives, scientists can trace the evolutionary changes that occurred over millions of years. The digestive systems of the theropod dinosaurs, for example, show remarkable similarities to those of modern birds, providing direct evidence of the evolutionary transition from dinosaurs to their feathered descendants. The muscle tissue preserved in these fossils offers insights into the biomechanics of extinct animals. By studying the arrangement and structure of ancient muscle fibers, researchers can reconstruct how these animals moved and estimate their strength and agility. This information helps bring these long-dead creatures back to life in our imagination, allowing us to envision them as dynamic, active animals rather than static museum displays. The blood vessel networks preserved in some fossils provide clues about the physiology and metabolism of ancient animals. The branching patterns of these vascular structures can reveal information about blood pressure, heart rate, and even body temperature regulation. Such details help scientists understand whether dinosaurs were warm-blooded or cold-blooded, a debate that has continued for decades and has important implications for our understanding of their behavior and ecology. As we continue our gentle journey through these remarkable discoveries, we begin to appreciate the extraordinary circumstances that must align to create such perfect preservation. The rapid burial that protects tissues from scavengers and bacterial decay. The specific chemical conditions that prevent normal decomposition while allowing mineralization to occur. The geological stability that preserves these delicate structures through millions of years of Earth's dynamic history. Each of these fossils represents a moment frozen in time, a snapshot of life from the distant past that provides insights unavailable through any other means. They remind us that the Earth has been home to countless extraordinary creatures, each adapted to their particular environment and way of life. The preservation of their soft tissues allows us to understand not just their anatomy, but their behavior, their ecology, and their place in the complex web of ancient life. The study of these exceptional fossils continues to reveal new secrets about the ancient world. As preservation techniques improve and new analytical methods are developed, scientists are able to extract ever more detailed information from these precious specimens. Each new discovery adds another piece to the puzzle of life's long history on Earth, helping us understand the evolutionary processes that have shaped the incredible diversity of life we see today. Tonight, as we rest and reflect on these remarkable discoveries, we can marvel at the incredible preservation that has allowed us to peer directly into the bodies of animals that lived millions of years ago. These fossils bridge the vast gulf of time that separates us from the ancient world, providing tangible connections to life forms that existed when our planet was young and very different from the world we know today. The preservation of these extraordinary specimens offers us glimpses into aspects of ancient life that would otherwise remain forever hidden. As we delve deeper into the mechanisms that created these fossilized treasures, we begin to understand just how remarkable the circumstances must have been for such delicate structures to survive the passage of geological time. Consider the complexity of the fossilization process itself. When an animal dies under normal conditions, decomposition begins almost immediately. Bacteria break down soft tissues within hours or days, leaving behind only the hardest parts like bones and teeth. But in the rare instances where these exceptional fossils formed, something interrupted this natural cycle of decay. The animals were buried so rapidly and completely that the usual processes of decomposition were halted before they could destroy the delicate internal structures. The sedimentary environments where these fossils formed were unlike typical burial sites. They required specific chemical conditions that could both prevent decay and facilitate the gradual replacement of organic materials with stable minerals. The water chemistry had to be just right, with the proper balance of acidity and mineral content to create an environment where soft tissues could be preserved rather than dissolved away. In the limestone quarries of Italy, the conditions that preserved our small theropod were likely created by a catastrophic event. Perhaps a sudden storm surge or underwater landslide buried the tiny predator in fine sediment before scavengers could disturb its remains. The alkaline environment of the ancient seafloor, combined with low oxygen levels, created the perfect conditions for phosphatization to occur. Over thousands of years, calcium phosphate minerals gradually replaced the original organic molecules, creating a perfect mineral replica of the animal's internal anatomy. The Chinese fossil beds tell a similar story, but with their own unique circumstances. Here, the preservation occurred in a freshwater lake environment where periodic volcanic activity may have contributed to the exceptional fossilization conditions. Fine volcanic ash settling through the water column could have provided the rapid burial necessary to protect the carcasses from decomposition, while the chemical composition of the ash itself may have contributed to the mineralization process. Each layer of sediment in these fossil deposits represents a moment in time, a snapshot of the ancient environment captured in stone. By studying these layers, scientists can reconstruct the climate, vegetation, and ecological conditions that existed when these animals lived. The fossils don't exist in isolation, but are part of a larger picture of ancient ecosystems that scientists are still working to understand. The preservation of feather impressions in the Chinese specimen adds another dimension to our understanding of these ancient creatures. Feathers are among the most delicate of all biological structures, consisting of fine keratin filaments that would normally decompose within days of death. Yet here they are, preserved in exquisite detail that reveals not just their presence, but their structure and arrangement. This preservation suggests that the burial was not only rapid, but also occurred in an environment where even the most fragile tissues could be protected. The blood vessels preserved in these fossils provide particularly intimate insights into the lives of these ancient animals. The circulatory system is the highway that carries nutrients and oxygen to every cell in the body, and its preservation allows us to understand aspects of physiology that would be impossible to deduce from bones alone. The branching patterns of these vessels reveal information about blood pressure, circulation efficiency, and even metabolic rates. When we examine the fossilized blood vessels under high magnification, we can see the incredible detail of their preservation. The smallest capillaries, vessels so thin that red blood cells must pass through them in single file, are visible as delicate traces threading through the mineralized tissues. These tiny structures once carried the life force of these ancient predators, delivering oxygen to their muscles as they hunted through prehistoric landscapes. The study of these vascular networks has revealed surprising similarities between dinosaur circulation and that of modern birds. The arrangement of blood vessels in the preserved organs suggests that these ancient theropods may have had more efficient circulatory systems than previously thought, supporting the theory that they were active, warm-blooded creatures rather than sluggish, cold-blooded reptiles. The muscle tissue preserved in these fossils tells its own story of ancient life. Under electron microscopy, individual muscle fibers are visible, some showing the characteristic striations that indicate they were capable of rapid, powerful contractions. The arrangement of these fibers provides clues about the animal's strength and agility, helping scientists understand how these predators moved through their environment. Different types of muscle tissue have been preserved in various parts of the fossils. The muscles of the neck region, crucial for the rapid head movements necessary for catching prey, show adaptations for both strength and speed. The preserved tail muscles reveal the complex arrangements needed to balance the animal's body during rapid turns and sudden stops. Even the fine muscles that control the movement of individual fingers and toes have left their traces in the fossil record. The preservation of these muscle structures has allowed scientists to create detailed reconstructions of how these ancient animals moved. By studying the attachment points of muscles to bones and the fiber arrangements within the muscles themselves, researchers can estimate the forces these creatures could generate and the speeds they could achieve. This biomechanical analysis has revealed that many theropod dinosaurs were far more athletic than previously imagined. The digestive systems preserved in these fossils provide perhaps the most direct evidence of how these ancient animals lived. The contents of their last meals, still visible within the fossilized intestines, tell stories of individual hunting events that occurred over 100 million years ago. Each prey item represents a moment of success for these ancient predators, a testament to their skill and adaptability. The preservation of stomach contents has revealed dietary patterns that were previously unknown. The small theropod from Italy, despite its youth, was already consuming a diverse array of prey, including both aquatic and terrestrial animals. This dietary flexibility suggests that these predators were opportunistic hunters capable of adapting their hunting strategies to whatever prey was available in their environment. The order in which the prey items were consumed, still visible in the fossilized digestive tract, provides insights into the animal's feeding behavior. The pattern suggests that this young predator was actively hunting multiple times per day, consuming prey items in quick succession. This high feeding frequency indicates a rapid metabolism and active lifestyle that required constant energy input. The absence of stomach tissue in these fossils, while initially puzzling, has provided its own insights into the preservation process. The highly acidic environment of the stomach continued to break down tissue even after death, preventing the preservation of this organ while allowing the less acidic intestines to be mineralized. This selective preservation helps scientists understand the chemical conditions that existed during fossilization. The respiratory systems preserved in these fossils offer glimpses into how these ancient animals breathed. The tracheal rings visible in the Italian specimen are remarkably similar to those found in modern birds, suggesting that the respiratory efficiency that allows birds to fly may have evolved much earlier than previously thought. These cartilaginous structures provided the flexibility needed for rapid breathing, while maintaining the structural integrity of the airway. The preservation of these delicate respiratory structures required exceptional conditions. Cartilage, being softer than bone but harder than pure soft tissue, occupies a middle ground in terms of preservation potential. The fact that these structures survived suggests that the fossilization process began very quickly after death, before the normal processes of decomposition could destroy these intermediate tissues. The brain preservation found in the English specimen represents the pinnacle of soft tissue fossilization. Neural tissue, with its high water content and lack of supporting structures, is among the most fragile of all biological materials. The fact that traces of actual brain matter have survived for over 100 million years demonstrates just how perfect the preservation conditions must have been. The chemical signature of this preserved brain tissue has revealed details about dinosaur neurochemistry that were previously thought to be forever lost. The iron-rich minerals found within the fossilized neural structures likely originated from hemoglobin in the blood that once flowed through the brain's extensive vascular network. This means that we may actually be detecting the chemical traces of thoughts and sensations that occurred in this ancient animal's mind. The organization of the preserved brain structures suggests that iguanodonts had more complex neural architecture than previously assumed. The folded surface of the brain, visible as faint impressions in the fossil, indicates a greater surface area for neural processing than would be expected from a simple, smooth brain. This complexity suggests that these herbivorous dinosaurs may have been capable of sophisticated behaviors, including complex social interactions and environmental problem-solving. The preservation of blood vessel networks within the brain tissue provides insights into the metabolic requirements of dinosaur neural tissue. The density and arrangement of these vessels suggest that dinosaur brains had high energy demands, similar to those of modern birds and mammals. This finding supports the theory that dinosaurs were active, intelligent creatures rather than sluggish, primitive reptiles. The study of these exceptional fossils has required the development of new analytical techniques and preservation methods. Each specimen is irreplaceable, representing millions of years of perfect preservation conditions that may never be replicated. Scientists must balance the need to extract maximum information from these fossils with the requirement to preserve them for future study as new technologies become available. Advanced imaging techniques have revolutionized the study of these specimens. High-resolution CT scanning allows researchers to examine internal structures without damaging the fossil, creating detailed three-dimensional maps of preserved organs and tissues. Synchrotron radiation generated by particle accelerators can reveal chemical compositions at the molecular level, detecting traces of original organic compounds that survived the fossilization process. Chemical analysis techniques have become increasingly sophisticated, allowing scientists to identify specific proteins, lipids, and even DNA-related molecules in these ancient specimens. While complete DNA extraction remains elusive, the detection of organic molecular fragments provides clues about the biochemistry of these extinct animals and their evolutionary relationships to modern species. The preservation of these soft tissues has also provided new insights into the environmental conditions that existed during the Cretaceous period. The types of bacteria and other microorganisms that contributed to the fossilization process can be identified through their chemical signatures, providing information about ancient ecosystems and climate conditions. The study of these fossils has revealed that soft tissue preservation may be more common than previously thought. As scientists have learned to recognize the subtle signs of preserved organic materials, they have begun to find traces of soft tissues in fossils that were previously thought to contain only mineral replacements. This discovery suggests that many museum specimens may contain hidden treasures of preserved biological materials waiting to be discovered. The implications of these discoveries extend far beyond paleontology. The preservation mechanisms that created these fossils are providing insights into how biological materials can be stabilized over geological time periods. This knowledge has applications in fields ranging from medicine to material science, where understanding long-term preservation of biological structures is crucial. The exceptional preservation of these fossils has also inspired new approaches to the study of modern animals. By understanding how ancient soft tissues were preserved, scientists can better interpret the anatomy and physiology of living species. The comparative approach, examining both fossil and modern organisms, provides a more complete picture of how complex biological systems have evolved over time. As we continue our peaceful journey through these remarkable discoveries, we can appreciate the dedication of the scientists who study these precious specimens. Each fossil represents years of careful preparation, analysis, and interpretation. The researchers who work with these materials must combine expertise in geology, biology, chemistry, and physics to unlock the secrets contained within these ancient treasures. The collaborative nature of this research brings together scientists from around the world, each contributing their unique expertise to understanding these complex specimens. Paleontologists, anatomists, biochemists, and imaging specialists work together to extract maximum information from each fossil, creating a comprehensive picture of ancient life that would be impossible for any single researcher to achieve alone. The preservation of these fossils in museum collections ensures that future generations of scientists will be able to study them with techniques that haven't been invented yet. Many of the most significant discoveries about these specimens have been made decades after their initial discovery, as new analytical methods revealed previously hidden details about their structure and composition. The public exhibition of these fossils serves an important educational function, allowing people to connect directly with the ancient past. When visitors see these preserved organs and tissues, they gain a visceral understanding of the reality of prehistoric life that goes beyond what can be conveyed through books or digital media. These fossils make the distant past tangible and immediate, inspiring wonder and curiosity about the natural world. The conservation of these specimens requires constant attention and specialized techniques. The minerals that replace the original organic materials are often unstable in museum environments, requiring careful control of temperature, humidity, and light exposure. Conservators must balance the need to preserve these materials for future study with the desire to make them accessible for research and education. The discovery of these exceptional fossils has also highlighted the importance of protecting fossil-bearing sites around the world. The specific conditions that created these remarkable specimens are rare and fragile, easily destroyed by human activity or natural processes. Conservation efforts must balance the need for scientific access with the protection of these irreplaceable resources. As we reflect on these extraordinary specimens in the quiet hours of the night, we can marvel at the chain of events that brought them to our attention. From the moment of death in ancient environments to their discovery by dedicated fossil hunters, each step in their journey required perfect timing and exceptional circumstances. These fossils represent not just scientific specimens, but miraculous survivals from a distant past that continues to inform our understanding of life on Earth. The study of these preserved organs and tissues reminds us that the boundary between past and present is not as absolute as we might imagine. The molecules that once functioned in these ancient animals have been transformed but not destroyed, creating a direct physical connection between our modern world and the distant past. When we examine these fossils, we are literally touching materials that once lived and breathed in ecosystems that existed millions of years ago. Tonight, as we rest and contemplate these remarkable discoveries, we can appreciate the incredible journey that has brought these ancient treasures to our attention. From their formation in prehistoric environments to their careful study by modern scientists, these fossils represent one of the most direct connections we have to the deep history of life on Earth. They remind us that our planet has been home to countless extraordinary creatures, each perfectly adapted to their particular time and place in the long story of evolution. The molecular signatures preserved within these ancient specimens tell stories that extend far beyond simple anatomical descriptions. When scientists analyze the chemical composition of these fossilized tissues, they discover traces of the very processes that once sustained life in these creatures. The iron-rich deposits found within the preserved organs represent more than just mineral replacements. They are the chemical echoes of blood that once flowed through living veins, carrying oxygen to muscles that powered movement through prehistoric landscapes. The preservation process itself created a kind of molecular archaeology, where each chemical signature provides clues about the original biochemistry of these ancient animals. The calcium phosphate minerals that replaced the organic tissues did so with such precision that they maintained not only the gross anatomy, but also the microscopic structure of cells and even subcellular components. This level of preservation allows scientists to study aspects of ancient physiology that bridge the gap between paleontology and modern biology. As we examine these specimens more closely, we begin to understand that their preservation required not just favorable conditions at the moment of death, but a sustained period of chemical stability that lasted for millions of years. The sedimentary environments where these fossils formed remained undisturbed by the geological upheavals that typically destroy such delicate preservation. Mountain building events, volcanic activity, and changes in groundwater chemistry all could have obliterated these precious records of ancient life. The rarity of such preservation becomes even more remarkable when we consider the vast number of animals that lived and died during the Cretaceous period. For every specimen that achieved this level of preservation, millions of others decomposed completely, leaving no trace of their existence. The fossils we study tonight represent not just individual animals, but lottery winners in the grand game of fossilization, specimens that beat astronomical odds to survive intact until their discovery by modern science. The intestinal contents preserved within these fossils provide particularly intimate glimpses into the daily lives of these ancient predators. The sequence of prey items found within the digestive tract reveals not just what these animals ate, but how they hunted and when they fed. The presence of both aquatic and terrestrial prey suggests that these predators were versatile hunters, capable of exploiting diverse food sources within their environment. The fish remains found within the fossilized stomach. Contents can be identified to species level, providing insights into the aquatic ecosystems that existed alongside these terrestrial predators. The preservation of scales, bones, and even soft tissues from these prey items creates a nested preservation phenomenon, where one exceptional fossil contains multiple examples of extraordinary preservation within its own body cavity. The lizard remains found intermixed with the fish provide evidence of the terrestrial hunting abilities of these ancient predators. The bones of these small reptiles are preserved with such clarity that paleontologists can identify specific species and even estimate the age and size of individual prey items. This level of detail allows scientists to reconstruct the hunting strategies and feeding behaviors of predators that lived over 100 million years ago. The order in which these prey items were consumed tells a story of active hunting behavior that contradicts older theories about dinosaur metabolism and activity levels. The rapid succession of feeding events preserved in these fossils suggests that these animals had high metabolic rates and required frequent meals to maintain their energy levels. This feeding pattern is more consistent with warm-blooded metabolism than with the sluggish, cold-blooded behavior once attributed to dinosaurs. The preservation of partially digested prey items within different sections of the intestinal tract provides insights into the digestive process itself. The degree of breakdown observed in different prey items indicates how long they had been in the digestive system at the time of death. This information allows scientists to estimate digestion rates and understand the efficiency of ancient digestive systems. The chemical analysis of these preserved stomach contents has revealed traces of digestive enzymes and stomach acids that were active at the time of death. These biochemical signatures provide direct evidence of the physiological processes that were occurring in these animals during their final hours. The preservation of such delicate biochemical information demonstrates just how perfect the fossilization conditions must have been. The muscular system preserved in these fossils reveals the sophisticated locomotory capabilities of these ancient predators. The arrangement of muscle fibers in the neck region shows adaptations for rapid head movements necessary for catching agile prey. The preserved muscle attachments indicate that these animals could generate significant bite forces, making them formidable hunters despite their relatively small size. The tail musculature preserved in these specimens demonstrates the complex arrangements needed for balance and maneuverability. The intricate network of muscles that controlled tail movement would have been essential for the rapid changes in direction required during pursuit of prey. The preservation of these soft tissue structures provides evidence for the athletic capabilities that made these predators successful in their ancient ecosystems. The respiratory systems preserved in these fossils offer insights into the metabolic demands of these active predators. The tracheal rings that supported the airway are remarkably similar to those found in modern birds, suggesting that efficient respiratory systems evolved early in theropod dinosaur evolution. This respiratory efficiency would have been crucial for sustaining the high activity levels indicated by other aspects of their anatomy. The preservation of lung tissue impressions in some specimens provides additional evidence for advanced respiratory systems. The structure of these preserved respiratory surfaces suggests that these animals had efficient gas exchange mechanisms that could support high metabolic rates. This finding supports theories about dinosaur warm-bloodedness and challenges older ideas about reptilian physiology. The cardiovascular system preserved in these fossils reveals the sophisticated circulatory adaptations needed to support active predatory behavior. The branching patterns of blood vessels show efficient distribution networks that could deliver oxygen and nutrients to rapidly working muscles. The preservation of these vascular structures provides direct evidence for the physiological capabilities of these ancient animals. The heart muscle tissue preserved in some specimens shows structural features consistent with high-performance cardiovascular systems. The arrangement of cardiac muscle fibers suggests that these animals had powerful hearts, capable of maintaining high blood pressure and rapid circulation. This cardiovascular efficiency would have been essential for supporting the energy demands of active hunting behavior. The nervous system preservation found in the English specimen represents perhaps the most remarkable aspect of these discoveries. The traces of actual brain tissue preserved within the fossilized skull provide unprecedented insights into the neural capabilities of these ancient herbivores. The complexity of the preserved neural structures suggests that these animals had more sophisticated cognitive abilities than previously assumed. The blood vessel networks preserved within the brain tissue indicate that these organs had high metabolic demands. The density of vascular structures suggests that dinosaur brains required significant energy input, similar to the energy-hungry brains of modern birds and mammals. This finding challenges older ideas about dinosaur intelligence and cognitive capabilities. The preservation of specific brain regions provides insights into the sensory and behavioral capabilities of these ancient animals. The areas of the brain associated with vision, hearing, and motor control show sophisticated organization that suggests complex behavioral repertoires. These neural adaptations would have been crucial for survival in competitive prehistoric ecosystems. The chemical signatures preserved within the brain tissue provide clues about the neurotransmitters and other biochemical factors that influence behavior in these ancient animals. The iron-rich deposits found within neural structures may represent the remains of hemoglobin from the extensive blood supply that supported brain function. This biochemical preservation offers insights into the physiological basis of ancient behavior. The comparative analysis of these brain fossils with modern animals reveals evolutionary continuities that span millions of years. The organization of neural structures shows similarities to both modern birds and reptiles, providing evidence for the evolutionary relationships between these groups. This comparative approach helps scientists understand how complex nervous systems evolved over geological time. The preservation of sensory organs within these fossils provides insights into how these ancient animals perceived their world. The structure of preserved eye tissues suggests sophisticated visual capabilities that would have been essential for hunting and navigation. The preservation of inner ear structures indicates advanced balance and spatial orientation systems. The social implications of these discoveries extend beyond individual anatomy to questions about group behavior and communication. The preservation of vocal structures in some specimens suggests that these animals may have been capable of complex acoustic communication. This finding has implications for understanding social behavior and group coordination in ancient ecosystems. The developmental insights provided by these fossils reveal how these complex anatomical systems emerged during growth and maturation. The juvenile specimens show intermediate stages in the development of adult characteristics, providing evidence for the evolutionary processes that shaped these remarkable creatures. This developmental perspective helps scientists understand how evolution produces complex biological systems. The ecological context of these discoveries provides insights into the environmental conditions that supported such sophisticated predators. The diversity of prey species preserved within these fossils indicates rich ecosystems with complex food webs. The preservation of both aquatic and terrestrial prey suggests that these predators operated at the interface between different habitat types. The seasonal patterns preserved in some specimens provide evidence for behavioral adaptations to environmental cycles. The timing of feeding events and the types of prey consumed suggest that these animals adjusted their hunting strategies in response to seasonal changes in prey availability. This behavioral flexibility would have been crucial for survival in dynamic prehistoric environments. The preservation of parasite remains within some fossils provides insights into the disease ecology of ancient ecosystems. The identification of fossilized parasites within the digestive tracts of these specimens reveals the complex relationships between predators and the microscopic organisms that affected their health. This perspective adds another dimension to our understanding of ancient ecological communities. The taphonomic processes that created these exceptional fossils continue to inform our understanding of how biological information can be preserved over geological time. The study of these preservation mechanisms has applications beyond paleontology, providing insights into how biological materials can be stabilized for long-term storage. This knowledge has implications for fields ranging from medicine to biotechnology. The analytical techniques used to study these fossils continue to evolve, revealing new details about their structure and composition. Advanced imaging methods allow scientists to examine these specimens at resolutions that were impossible just a few years ago. Each technological advance brings new insights about these ancient animals and the processes that preserve them. The collaborative nature of modern paleontological research brings together expertise from multiple disciplines to study these complex specimens. Geologists, biologists, chemists, and physicists work together to extract maximum information from each fossil, creating comprehensive pictures of ancient life that would be impossible for any single researcher to achieve alone. The educational value of these discoveries extends far beyond the scientific community. When students and the public see these preserved organs and tissues, they gain a visceral understanding of the reality of prehistoric life that goes beyond abstract concepts. These fossils make the deep past tangible and immediate, inspiring curiosity about the natural world and the processes that have shaped life on Earth. The conservation challenges associated with these specimens require constant attention and specialized expertise. The minerals that replace the original organic materials are often unstable in museum environments, requiring careful control of storage conditions. Conservators must balance the need to preserve these materials for future study with the desire to make them accessible for research and education. The discovery of these exceptional fossils has also highlighted the importance of protecting fossil-bearing sites around the world. The specific conditions that created these remarkable specimens are rare and fragile, easily destroyed by human activity or natural processes. Conservation efforts must balance the need for scientific access with the protection of these irreplaceable resources for future generations. As we continue our quiet journey through these remarkable discoveries, we can appreciate the dedication of the amateur fossil collectors who made many of these finds possible. Their careful observation and patient preparation work often provides the first glimpse of these extraordinary specimens. The collaboration between amateur collectors and professional scientists has been crucial for advancing our understanding of these ancient treasures. The preparation techniques used to reveal these fossils require exceptional skill and patience. The delicate work of removing the surrounding rock while preserving microscopic details of soft tissue can take months or even years to complete. The preparators who perform this work must combine artistic sensitivity with scientific precision to reveal the full extent of these remarkable preservations. The photographic documentation of these specimens creates permanent records that can be shared with researchers around the world. The specialized lighting and imaging techniques used to capture the subtle details of preserved soft tissues require expertise in both paleontology and photography. These images become valuable scientific resources that can be studied and re-examined for decades after their creation. The digital modeling techniques used to study these fossils allow scientists to create three-dimensional reconstructions of ancient anatomy. These models can be manipulated and analyzed in ways that would be impossible with the original specimens, providing new insights into the structure and function of ancient biological systems. The digital preservation of these models ensures that this information will be available for future research. The molecular analysis of these specimens continues to reveal new aspects of their preservation and composition. The detection of organic compounds within fossilized tissues provides insights into the biochemistry of these ancient animals and the processes that preserve them. These molecular signatures create bridges between the ancient and modern worlds, connecting us directly to the biological processes that sustained life millions of years ago. The isotopic analysis of these fossils provides information about the environments in which these animals lived and died. The ratios of different isotopes preserved within the fossilized tissues can reveal details about ancient climate conditions, food webs, and even migration patterns. This environmental context helps scientists understand the ecological circumstances that shape the evolution of these remarkable creatures. The comparative studies that place these fossils in evolutionary context reveal the long history of complex biological systems on Earth. The similarities between ancient and modern anatomical structures demonstrate the continuity of life processes across geological time. These comparisons help scientists understand how evolution has modified and refined biological systems over millions of years. The biomechanical analysis of these preserved structures provides insights into how these ancient animals moved and functioned. The arrangement of muscle fibers, the structure of joints, and the organization of support systems all provide clues about the capabilities and limitations of these extinct creatures. This functional analysis helps bring these ancient animals to life in our imagination. The behavioral implications of these discoveries extend our understanding of these animals beyond simple anatomical descriptions. The preservation of feeding behavior, digestive processes, and even neural structures provides insights into the complex behaviors that allowed these creatures to survive and thrive in their ancient ecosystems. These behavioral reconstructions help us understand these animals as living, breathing creatures rather than static museum displays. The evolutionary significance of these discoveries continues to reshape our understanding of the transition from dinosaurs to modern birds. The preservation of respiratory systems, cardiovascular structures, and neural organizations provides direct evidence for the evolutionary changes that occurred during this important transition. These fossils serve as evolutionary snapshots that capture intermediate stages in the development of modern biological systems. The technological innovations inspired by these discoveries have applications far beyond paleontology. The preservation mechanisms that created these fossils are providing insights into how biological materials can be stabilized over long time periods. This knowledge has implications for medical device development, material science, and biotechnology applications. The philosophical implications of these discoveries touch on fundamental questions about the nature of life and time. The preservation of thoughts and sensations from millions of years ago challenges our understanding of the permanence of consciousness and experience. These fossils create tangible connections between past and present that transcend the usual boundaries of scientific inquiry. As we rest in the quiet hours of the night, contemplating these extraordinary windows into the ancient past, we can marvel at the remarkable preservation that has allowed us to study the most intimate details of creatures that lived in worlds vastly different from our own. These fossils represent more than scientific specimens. They are time capsules that have survived impossible odds to deliver messages from the deep past about the nature of life on Earth. The study of these preserved organs and tissues reminds us that the history of life on our planet is far richer and more complex than we might imagine from bones alone. The soft tissues that once animated these ancient creatures have left their traces in the rock record, creating opportunities for understanding that extend far beyond traditional paleontological approaches. These discoveries continue to reveal new secrets about the ancient world, each specimen adding another piece to the vast puzzle of life's history. Tonight, as we continue our peaceful exploration of these remarkable fossils, we can appreciate the incredible journey that has brought these ancient treasures to our attention. From their formation in prehistoric environments to their discovery by dedicated fossil hunters, each step in their preservation required perfect timing and exceptional circumstances. These specimens represent miraculous survivals from distant worlds that continue to inform our understanding of life's long history on Earth. The legacy of these discoveries extends far beyond the scientific papers and museum displays they have inspired. They represent humanity's ongoing quest to understand our place in the vast sweep of natural history, connecting us to the deep roots of life on Earth and reminding us that we are part of a story that began billions of years ago and continues to unfold around us today. The molecular clock embedded within these ancient specimens tells a story that extends far beyond the visible structures we can observe. When scientists examine the chemical composition of these fossilized tissues at the atomic level, they discover that the mineralization process occurred with such precision that it captured not just the shape of cells, but the very arrangement of molecules that once sustained life. The phosphate crystals that replaced the original organic compounds did so in patterns that mirror the biological organization of living tissue. This molecular preservation creates a kind of chemical fingerprint that persists across geological time. The iron signatures found within the preserved blood vessels represent more than just mineral deposits. They are the transformed remains of hemoglobin molecules that once carried oxygen through living circulatory systems. The calcium compounds that replaced bone and cartilage maintain the microscopic structure of the original materials, preserving details down to the level of individual bone cells. The preservation process itself reveals the delicate balance of conditions required for such extraordinary fossilization. The pH levels of the ancient environments where these specimens formed had to remain stable for thousands of years during the gradual replacement of organic materials. Too acidic, and the delicate tissues would have dissolved completely. Too alkaline, and the mineralization process would have been disrupted, leaving only crude impressions rather than the exquisite detail we observe today. The temperature stability of these ancient burial environments was equally crucial. Fluctuations in temperature would have caused expansion and contraction of the surrounding sediments, potentially destroying the delicate three-dimensional relationships between preserved structures. The consistent cool temperatures that existed in these deep burial environments provided the thermal stability necessary for such precise preservation. The water chemistry of these ancient environments played a particularly important role in determining which tissues would be preserved and which would be lost. The dissolved minerals in the groundwater that percolated through these burial sites provided the raw materials for fossilization, while the pH and ionic composition of these solutions determined how the replacement process would proceed. Different minerals were deposited at different rates, creating the layered preservation we observe in these specimens. The bacterial communities that existed in these ancient burial environments contributed to the preservation process in ways that scientists are only beginning to understand. Certain types of bacteria can create chemical conditions that promote mineralization, while others can produce compounds that inhibit decay. The balance between these microbial communities may have been crucial in determining which specimens achieved exceptional preservation. The study of these bacterial contributions to fossilization has revealed that preservation was not simply a passive process of mineral replacement. Instead, it was an active biochemical process involving complex interactions between organic decay products, mineral-rich groundwater, and microbial metabolism. This understanding has changed how scientists think about fossilization and has led to new insights about the conditions necessary for exceptional preservation. The oxygen levels in these ancient burial environments were carefully balanced to promote preservation while preventing complete decomposition. Too much oxygen would have allowed aerobic bacteria to completely break down the soft tissues before mineralization could occur. Too little oxygen would have prevented the chemical reactions necessary for the formation of stable mineral replacements. The environments that created these fossils maintained just the right balance of oxygen availability. The sediment grain size in these burial environments was another crucial factor in preservation quality. Fine-grained sediments like clay and silt could pack closely around the specimens, excluding oxygen and creating the anaerobic conditions necessary for preservation. Coarse-grained sediments would have allowed too much water circulation, disrupting the delicate chemical conditions required for soft tissue fossilization. The rate of burial was perhaps the most critical factor in determining preservation quality. Specimens that were buried slowly would have been exposed to scavengers and decay processes that would have destroyed soft tissues before fossilization could begin. Only specimens that were buried rapidly, perhaps by sudden sediment flows or volcanic ash falls, had the opportunity to achieve the exceptional preservation we observe in these fossils. The geological stability of these burial sites over millions of years was essential for maintaining the integrity of these delicate fossils. Tectonic activity that folded or fractured the surrounding rock layers would have destroyed the preserved soft tissues through compression and shearing. The fossils we study today survived in areas that remained geologically quiet for vast periods of time. The groundwater systems that existed in these ancient environments continued to influence preservation long after the initial burial. The flow of mineral-rich water through the surrounding sediments provided the dissolved compounds necessary for continued mineralization. Changes in groundwater chemistry over time could either enhance preservation or begin to dissolve previously formed minerals. The depth of burial affected preservation in complex ways. Shallow burial exposed specimens to temperature fluctuations and chemical changes that could disrupt fossilization. Deep burial provided stable conditions but increased pressure that could compress and distort delicate structures. The optimal depth for preservation balanced these competing factors to maintain both chemical stability and structural integrity. The surrounding rock chemistry influenced preservation through its effects on groundwater composition. Limestone environments, rich in calcium carbonate, promoted the formation of calcium phosphate minerals that replaced soft tissues. Sandstone environments, with their different mineral compositions, created different preservation conditions that might favor some tissues over others. The time scales involved in these preservation processes are almost incomprehensible. The gradual replacement of organic molecules with stable minerals occurred over thousands of years, proceeding at rates so slow that individual chemical reactions took place over decades. This incredibly slow pace allowed for the precise molecular replacement that created such detailed preservation. The preservation of cellular structures within these fossils provides insights into the microscopic organization of ancient life. Individual cells are visible in some specimens, their boundaries marked by subtle differences in mineral composition. The preservation of cell walls, nuclei, and even organelles creates opportunities to study ancient life at levels of detail that approach modern biological research. The chemical gradients preserved within these fossils reveal the metabolic processes that were active at the time of death. The distribution of different elements throughout the preserved tissues reflects the biochemical pathways that were transporting nutrients and waste products through these ancient bodies. These chemical signatures provide direct evidence of the physiological processes that sustained life millions of years ago. The preservation of enzyme activity signatures within these fossils represents perhaps the most remarkable aspect of their chemistry. The specific arrangements of mineral crystals in certain regions suggest that they formed in response to the catalytic activity of biological enzymes. This means that the actual biochemical processes of life may have influenced the fossilization process, creating mineral patterns that reflect ancient metabolic activity. The isotopic signatures preserved within these fossils provide information about the environmental conditions that existed during the lives of these ancient animals. The ratios of different isotopes in the preserved tissues reflect the temperature, humidity, and chemical composition of the ancient atmospheres in which these creatures lived. This environmental information helps scientists reconstruct the ecological context that shaped the evolution of these remarkable species. The preservation of lipid signatures within these fossils has revealed traces of the fats and oils that once formed cell membranes and energy storage compounds. These organic molecules, while chemically altered by the fossilization process, retain enough of their original structure to provide insights into the biochemistry of ancient life. The detection of these compounds represents a direct molecular connection to the metabolic processes of creatures that lived millions of years ago. The protein signatures preserved within these fossils provide evidence for the molecular machinery that once drove cellular processes. While complete proteins have not survived the fossilization process, fragments and chemical traces of these complex molecules can still be detected using advanced analytical techniques. These molecular fossils provide insights into the evolutionary history of fundamental biological processes. The preservation of nucleic acid signatures within these fossils remains one of the most controversial aspects of their study. While complete DNA extraction from such ancient specimens remains highly unlikely, some researchers have reported detecting chemical traces that may represent degraded fragments of genetic material. These findings, while disputed, raise tantalizing possibilities about the molecular information that might be preserved in exceptional fossils. The collagen signatures preserved within these fossils provide insights into the structural proteins that once supported the bodies of these ancient creatures. Collagen being one of the most stable proteins in living systems, has the best chance of leaving detectable traces in the fossil record. The chemical analysis of these protein remnants provides information about the biomechanical properties of ancient tissues. The pigment signatures preserved within these fossils have revealed traces of the compounds that once gave these ancient animals their colors. The chemical analysis of fossilized feathers and skin has detected melanin-related compounds that provide clues about the coloration patterns of creatures that lived millions of years ago. This information helps scientists understand the visual appearance of these ancient animals and the ecological roles that coloration played in their lives. The preservation of sensory protein signatures within these fossils provides insights into how these ancient animals perceived their world. The chemical traces of visual pigments in preserved eye tissues suggest the wavelengths of light that these creatures could detect. Similarly, the analysis of inner ear structures has revealed traces of proteins associated with balance and hearing, providing clues about the sensory capabilities of these extinct species. The immune system's signatures preserved within these fossils provide evidence for the defense mechanisms that protected these ancient animals from disease. The chemical analysis of preserved lymphatic tissues has revealed traces of proteins associated with immune responses, suggesting that these creatures had sophisticated systems for fighting infection and maintaining health. The hormonal signatures preserved within these fossils provide insights into the regulatory systems that controlled growth, reproduction, and behavior in these ancient animals. The detection of steroid hormone metabolites in preserved tissues suggests that these creatures had complex endocrine systems similar to those found in modern vertebrates. These chemical traces provide evidence for the physiological processes that controlled the development and behavior of these extinct species. The neurotransmitter signatures preserved within brain tissues provide the most direct evidence yet discovered for the biochemical basis of ancient behavior. The chemical analysis of preserved neural structures has revealed traces of compounds associated with neurotransmission, suggesting that these ancient brains operated using chemical signaling systems similar to those found in modern animals. This finding provides unprecedented insights into the physiological basis of behavior in extinct species. The preservation of metabolic signatures within these fossils reveals the energy production systems that powered these ancient creatures. The chemical analysis of preserved muscle tissues has detected traces of compounds associated with cellular respiration and energy metabolism. These signatures provide evidence for the metabolic rates and energy requirements of these extinct animals. The developmental signatures preserved within juvenile specimens provide insights into the growth processes that shaped these ancient creatures. The chemical analysis of preserved growth plates and developing organs reveals traces of the proteins and hormones that control development. These signatures provide evidence for the developmental programs that guided the transformation from embryo to adult. In these extinct species, the seasonal signatures preserved within these fossils provide evidence for the cyclical processes that influenced the lives of these ancient animals. The chemical analysis of preserved tissues has revealed traces of compounds that vary with seasonal changes in diet, activity, and reproductive status. These signatures provide insights into the annual cycles that structured the lives of these extinct creatures. The stress signatures preserved within these fossils provide evidence for the challenges that these ancient animals faced in their daily lives. The chemical analysis of preserved tissues has revealed traces of stress hormones and other compounds associated with physiological responses to environmental challenges. These signatures provide insights into the survival pressures that shape the evolution of these extinct species. The social signatures preserved within these fossils provide evidence for the complex behaviors that may have characterized these ancient animals. The chemical analysis of preserved scent glands and other specialized tissues has revealed traces of compounds associated with communication and social interaction. These signatures suggest that these extinct creatures may have had sophisticated social systems similar to those found in modern animals. The reproductive signatures preserved within these fossils provide insights into the breeding behaviors and life cycles of these ancient creatures. The chemical analysis of preserved reproductive tissues has revealed traces of hormones and other compounds associated with mating and parental care. These signatures provide evidence for the reproductive strategies that allowed these species to survive and thrive in their ancient environments. The preservation of circadian signatures within these fossils provides evidence for the daily rhythms that structured the lives of these ancient animals. The chemical analysis of preserved tissues has revealed traces of compounds associated with biological clocks and daily activity patterns. These signatures provide insights into the temporal organization of behavior in these extinct species. The preservation of migration signatures within these fossils provides evidence for the long-distance movements that may have characterized some of these ancient species. The chemical analysis of preserved tissues has revealed isotopic signatures that suggest seasonal movements between different environments. These signatures provide insights into the mobility and habitat use patterns of these extinct creatures. The preservation of dietary signatures within these fossils provides detailed information about the feeding behaviors and ecological roles of these ancient animals. The chemical analysis of preserved digestive tissues has revealed traces of specific food compounds that provide insights into diet composition and feeding strategies. These signatures help scientists understand the ecological relationships that structured ancient food webs. The preservation of disease signatures within these fossils provides evidence for the health challenges that affected these ancient animals. The chemical analysis of preserved tissues has revealed traces of compounds associated with infection, injury, and other pathological conditions. These signatures provide insights into the disease ecology of ancient ecosystems and the evolutionary pressures that shaped immune system development. The preservation of toxin signatures within these fossils provides evidence for the chemical defenses that may have protected these ancient animals from predation. The chemical analysis of preserved tissues has revealed traces of compounds that may have made these creatures unpalatable or dangerous to potential predators. These signatures provide insights into the co-evolutionary arms races that shaped predator-prey relationships in ancient ecosystems. The preservation of thermal signatures within these fossils provides evidence for the temperature regulation systems that allowed these ancient animals to survive in their environments. The chemical analysis of preserved tissues has revealed traces of compounds associated with thermal regulation and metabolic heat production. These signatures provide insights into the physiological adaptations that allowed these creatures to thrive in ancient climates. The preservation of hydration signatures within these fossils provides evidence for the water balance systems that maintained physiological function in these ancient animals. The chemical analysis of preserved tissues has revealed traces of compounds associated with water regulation and osmoregulation. These signatures provide insights into the physiological challenges of maintaining water balance in ancient environments. The preservation of pH signatures within these fossils provides evidence for the acid-base regulation systems that maintained cellular function in these ancient creatures. The chemical analysis of preserved tissues has revealed traces of compounds associated with pH buffering and regulation. These signatures provide insights into the biochemical systems that maintained physiological stability in these extinct species. The preservation of oxygen signatures within these fossils provides evidence for the respiratory systems that delivered oxygen to the tissues of these ancient animals. The chemical analysis of preserved respiratory structures has revealed traces of compounds associated with oxygen transport and utilization. These signatures provide insights into the metabolic capabilities and activity levels of these extinct creatures. The preservation of carbon signatures within these fossils provides evidence for the carbon cycle processes that supported life in these ancient ecosystems. The chemical analysis of preserved tissues has revealed isotopic signatures that reflect the carbon sources and cycling processes that sustained these ancient food webs. These signatures provide insights into the biogeochemical processes that supported life in ancient environments. The preservation of nitrogen signatures within these fossils provides evidence for the protein synthesis and waste processing systems that maintain cellular function in these ancient animals. The chemical analysis of preserved tissues has revealed traces of compounds associated with nitrogen metabolism and excretion. These signatures provide insights into the biochemical processes that supported growth and maintenance in these extinct species. The preservation of phosphorus signatures within these fossils provides evidence for the energy storage and transfer systems that powered cellular processes in these ancient creatures. The chemical analysis of preserved tissues has revealed traces of compounds associated with phosphate metabolism and energy transfer. These signatures provide insights into the biochemical systems that supported life in these extinct species. The preservation of sulfur signatures within these fossils provides evidence for the protein structure and enzyme function systems that maintain cellular organization in these ancient animals. The chemical analysis of preserved tissues has revealed traces of compounds associated with sulfur metabolism and protein structure. These signatures provide insights into the molecular systems that supported life in these extinct creatures. The preservation of trace element signatures within these fossils provides evidence for the micronutrient systems that supported specialized cellular functions in these ancient animals. The chemical analysis of preserved tissues has revealed traces of elements like iron, copper, and zinc that were essential for enzyme function and cellular metabolism. These signatures provide insights into the nutritional requirements and metabolic capabilities of these extinct species. The integration of all of these preserved signatures creates a comprehensive picture of ancient life that extends far beyond traditional paleontological approaches. The combination of anatomical, chemical, and molecular evidence provides insights into the complete biology of these extinct creatures, from their cellular metabolism to their ecosystem interactions. This multidisciplinary approach represents a new frontier in paleontology that promises to reveal ever more detailed information about the history of life on Earth. The technological advances that have made these discoveries possible continue to push the boundaries of what can be learned from fossil specimens. New analytical techniques are constantly being developed that can detect ever more subtle traces of ancient biological processes. The application of these technologies to existing museum specimens is revealing hidden treasures of biological information that were previously invisible to scientific investigation. The collaborative nature of this research brings together scientists from diverse fields including paleontology, biochemistry, material science, and molecular biology. This interdisciplinary approach is essential for understanding the complex processes that created these remarkable fossils and for extracting maximum information from these precious specimens. The insights gained from these collaborations are advancing our understanding of both ancient life and the processes that govern biological systems. The implications of these discoveries extend far beyond the study of extinct species. The preservation mechanisms revealed by these fossils are providing insights into how biological materials can be stabilized over long time periods, with applications in medicine, biotechnology, and material science. The understanding of ancient biochemical processes is informing research into the evolution of fundamental biological systems and the origin of life itself. The educational value of these discoveries cannot be overstated. When students and the public learn about these preserved molecular signatures, they gain a deeper appreciation for the continuity of life processes across geological time. These findings make the connection between ancient and modern life tangible and immediate, inspiring curiosity about the natural world and the processes that have shaped life on Earth. The conservation challenges associated with these specimens require constant attention and specialized expertise. The preservation of the chemical information contained within these fossils is even more delicate than the preservation of their physical structures. Environmental factors that might not affect the gross anatomy of these specimens can nevertheless destroy the molecular signatures that provide the most detailed information about ancient life. The future of this research depends on continued technological advancement and interdisciplinary collaboration. As analytical techniques become more sensitive and less destructive, Scientists will be able to extract ever more detailed information from these precious specimens. The development of new approaches to studying ancient biological materials promises to reveal aspects of prehistoric life that we can barely imagine today. The philosophical implications of these discoveries touch on fundamental questions about the nature of biological information and its persistence through time. The preservation of molecular signatures from ancient life forms challenges our understanding of the boundaries between past and present, suggesting that the information content of life may be more persistent than we previously imagined. These fossils create direct molecular connections between ancient and modern life that transcend the usual boundaries of scientific inquiry. As we continue our peaceful exploration of these remarkable discoveries, we can appreciate the incredible chain of events that has preserved these molecular signatures across vast spans of geological time. The precise conditions that allowed these chemical traces to survive represent a perfect alignment of environmental factors that occurs perhaps once in millions of fossilization events. These specimens represent not just scientific curiosities, but miraculous survivals that provide unprecedented insights into the deep history of life on Earth. The study of these preserved molecular signatures reminds us that the history of life is written not just in stone, but in the very atoms and molecules that once sustained ancient creatures. These chemical traces create bridges across time that connect us directly to the biological processes that animated life millions of years ago. When we analyze these ancient molecular signatures, we are literally touching the transformed remains of the biochemical machinery that once powered movement, growth, and sensation in creatures that lived in worlds vastly different from our own. Tonight, as we rest and reflect on these extraordinary discoveries, we can marvel at the remarkable preservation processes that have allowed us to study the most intimate molecular details of creatures that lived during the age of dinosaurs. These fossils represent more than scientific specimens. They are chemical time capsules that have survived impossible odds to deliver messages from the deep past about the fundamental nature of life itself. The molecular signatures preserved within these ancient specimens continue to reveal new secrets about the biochemical processes that have sustained life on Earth for billions of years, creating connections between past and present that remind us of our place in the vast continuum of life's history. The preservation of these molecular signatures has opened entirely new avenues for understanding the daily lives of these ancient creatures. When scientists examine the chemical traces left behind in fossilized muscle tissue, they can detect patterns that reveal not just the structure of these tissues, but the very processes that once powered movement through prehistoric landscapes. The arrangement of protein fragments within these preserved fibers tells stories of contraction and relaxation, of the coordinated muscle activities that allowed these predators to pursue their prey with remarkable efficiency. The metabolic signatures embedded within these ancient tissues provide insights into the energy systems that sustained these creatures during their most active moments. The chemical traces of cellular respiration processes reveal that these animals had sophisticated energy production systems capable of supporting rapid, sustained activity. The preservation of these metabolic indicators challenges older assumptions about the activity levels of prehistoric creatures, suggesting that many possessed the physiological capabilities for dynamic, energetic lifestyles. The blood chemistry preserved within these specimens tells particularly intimate stories about the final moments of these ancient lives. The iron-rich deposits found throughout the fossilized circulatory systems represent more than simple mineral replacements. They are the chemical echoes of hemoglobin molecules that once carried oxygen through living bloodstreams, delivering the breath of life to every cell in these creatures' bodies. When researchers analyze these iron signatures, they are literally studying the transformed remains of the life force that once animated these prehistoric predators. The preservation of stress response signatures within these fossils provides evidence for the physiological challenges these animals faced in their daily existence. The chemical traces of stress hormones and related compounds suggest that these creatures experienced the same types of physiological responses to danger and challenge that characterize modern animals. These signatures provide insights into the survival pressures that shape the evolution of these remarkable species, revealing that the struggle for existence has remained fundamentally unchanged across millions of years. The digestive enzyme signatures preserved within the fossilized gut contents reveal the sophisticated biochemical processes that broke down prey items into usable nutrients. The chemical analysis of these preserved digestive systems has detected traces of proteases, lipases, and other enzymes that were actively processing food at the time of death. This biochemical evidence provides direct insights into the digestive capabilities of these ancient predators, suggesting that they possessed highly efficient systems for extracting maximum nutrition from their prey. The preservation of growth factor signatures within juvenile specimens provides unprecedented insights into the developmental processes that shaped these ancient creatures. The chemical traces of hormones and proteins associated with growth and development reveal that these animals underwent complex maturation processes, similar to those seen in modern vertebrates. These signatures provide evidence for the regulatory systems that controlled the transformation from hatchling to adult in these extinct species. The neural chemistry preserved within brain tissues represents perhaps the most remarkable aspect of these molecular discoveries. The chemical analysis of fossilized neural structures has revealed traces of neurotransmitters and related compounds that once facilitated communication between nerve cells. These molecular signatures provide direct evidence for the biochemical processes that once generated thoughts, sensations, and behaviors in these ancient minds. When scientists study these preserved neurochemicals, they are examining the transformed remains of the very molecules that once created consciousness in creatures that lived millions of years ago. The preservation of sensory molecule signatures within specialized tissues provides insights into how these ancient animals perceived their world. The chemical analysis of fossilized eye tissues has revealed traces of visual pigments that once detected light and color. Similarly, the study of preserved inner ear structures has identified molecular signatures associated with balance and hearing. These findings suggest that these creatures possessed sophisticated sensory systems that allowed them to navigate complex environments and detect both prey and predators with remarkable precision. The immune system signatures preserved within these fossils reveal the defensive mechanisms that protected these ancient animals from disease and injury. The chemical analysis of preserved lymphatic tissues has detected traces of antibodies and other immune system proteins that once fought infections and maintained health. These molecular signatures provide evidence for the sophisticated defense systems that allowed these creatures to survive in environments filled with pathogens and parasites. The reproductive hormone signatures preserved within these specimens provide insights into the breeding behaviors and life cycles of these ancient species. The chemical analysis of preserved reproductive tissues has revealed traces of hormones associated with mating displays, territorial behavior, and parental care. These signatures suggest that these creatures had complex reproductive strategies that involved sophisticated behaviors and physiological adaptations. The seasonal variation signatures preserved within these fossils provide evidence for the cyclical processes that structured the lives of these ancient animals. The chemical analysis of preserved tissues has revealed molecular traces that vary with seasonal changes in diet, activity levels, and reproductive status. These signatures indicate that these creatures were well adapted to the rhythmic changes in their environments, adjusting their physiology and behavior in response to seasonal cues. The social communication signatures preserved within specialized glands and tissues provide evidence for the complex interactions that may have characterized these ancient species. The chemical analysis of preserved scent-producing structures has revealed traces of pheromones and other signaling molecules that once facilitated communication between individuals. These molecular signatures suggest that these creatures had sophisticated social systems involving chemical communication, territorial marking, and group coordination. The thermal regulation signatures preserved within these fossils provide insight into the temperature control systems that allowed these ancient animals to maintain optimal body temperatures. The chemical analysis of preserved tissues has revealed traces of compounds associated with heat production, heat conservation, and temperature sensing. These signatures provide evidence for the physiological adaptations that allowed these creatures to thrive in the warm, humid climates of their ancient world. The circadian rhythm signatures preserved within these specimens provide evidence for the biological clocks that structured the daily activities of these ancient creatures. The chemical analysis of preserved tissues has revealed molecular traces of proteins associated with circadian regulation, suggesting that these animals had sophisticated timing systems that coordinated their physiology and behavior with daily cycles of light and darkness. The migration signatures preserved within these fossils provide evidence for the long-distance movements that may have characterized some of these ancient species. The isotopic analysis of preserved tissues has revealed chemical signatures that suggest seasonal movements between different environments. These signatures indicate that some of these creatures may have undertaken regular migrations in response to seasonal changes in food availability or breeding requirements. The territorial behavior signatures preserved within these specimens provide insights into the spatial organization of these ancient communities. The chemical analysis of preserved marking glands has revealed traces of compounds associated with territorial displays and boundary marking. These molecular signatures suggest that these creatures had complex systems for organizing their use of space and resources. The predator-prey interaction signatures preserved within these fossils provide evidence for the complex relationships that structured ancient food webs. The chemical analysis of preserved digestive contents has revealed molecular traces of specific prey species, providing insights into the feeding relationships that connected different levels of these ancient ecosystems. These signatures help scientists understand the ecological roles that these predators played in their prehistoric communities. The disease-resistant signatures preserved within these specimens provide evidence for the evolutionary pressures that shaped immune system development in these ancient lineages. The chemical analysis of preserved tissues has revealed traces of compounds associated with pathogen resistance and disease recovery. These signatures provide insights into the health challenges that these creatures faced and the evolutionary adaptations that allowed them to survive in environments filled with infectious agents. The toxin-resistant signatures preserved within these fossils provide evidence for the chemical defenses that may have protected these ancient animals from poisonous prey or environmental toxins. The chemical analysis of preserved liver and kidney tissues has revealed traces of detoxification enzymes and related compounds. These signatures suggest that these creatures had sophisticated systems for dealing with harmful chemicals in their environment. The water balance signatures preserved within these specimens provide insights into the osmoregulatory systems that maintained proper hydration in these ancient animals. The chemical analysis of preserved kidney tissues has revealed traces of compounds associated with water conservation and salt regulation. These signatures provide evidence for the physiological adaptations that allowed these creatures to maintain proper fluid balance in their ancient environments. The oxygen transport signatures preserved within these fossils provide evidence for the respiratory system that delivered oxygen to the tissues of these ancient creatures. The chemical analysis of preserved blood and respiratory tissues has revealed traces of oxygen-carrying proteins and related compounds. These signatures provide insights into the metabolic capabilities and activity levels of these extinct species. The waste processing signatures preserved within these specimens provide evidence for the excretory systems that maintained cellular health in these ancient animals. The chemical analysis of preserved kidney and liver tissues has revealed traces of compounds associated with waste filtration and elimination. These signatures provide insights into the metabolic processes that supported life in these extinct creatures. The pH regulation signatures preserved within these fossils provide evidence for the acid-base balance systems that maintained cellular function in these ancient species. The chemical analysis of preserved tissues has revealed traces of buffering compounds and pH-regulating proteins. These signatures provide insights into the biochemical systems that maintained physiological stability in these extinct animals. The mineral transport signatures preserved within these specimens provide evidence for the systems that delivered essential elements to the tissues of these ancient creatures. The chemical analysis of preserved tissues has revealed traces of transport proteins and mineral-binding compounds. These signatures provide insights into the nutritional requirements and metabolic capabilities of these extinct species. The energy storage signatures preserved within these fossils provide evidence for the systems that maintained energy reserves in these ancient animals. The chemical analysis of preserved fat and muscle tissues has revealed traces of compounds associated with energy storage and mobilization. These signatures provide insights into the metabolic strategies that allowed these creatures to survive periods of food scarcity. The cellular repair signatures preserved within these specimens provide evidence for the maintenance systems that kept these ancient animals healthy and functional. The chemical analysis of preserved tissues has revealed traces of repair proteins and cellular maintenance compounds. These signatures provide insights into the longevity and health maintenance strategies of these extinct species. The developmental timing signatures preserved within these fossils provide evidence for the regulatory systems that controlled growth and maturation in these ancient creatures. The chemical analysis of preserved tissues has revealed traces of developmental hormones and timing proteins. These signatures provide insights into the life history strategies that characterized these extinct species. The environmental adaptation signatures preserved within these specimens provide evidence for the physiological flexibility that allowed these ancient animals to survive in changing conditions. The chemical analysis of preserved tissues has revealed traces of stress response proteins and adaptation compounds. These signatures provide insights into the evolutionary strategies that allowed these creatures to thrive in dynamic prehistoric environments. The preservation of these molecular signatures represents more than just scientific curiosities. They provide direct evidence for the biochemical processes that sustained life in these ancient creatures, creating molecular bridges that connect us to the deep history of life on Earth. When scientists analyze these chemical traces, they are studying the transformed remains of the very molecules that once powered movement, growth, and sensation in animals that lived during the age of dinosaurs. The analytical techniques required to study these molecular signatures continue to advance at a rapid pace. New methods for detecting and analyzing ancient biological compounds are constantly being developed, allowing scientists to extract ever more detailed information from these precious specimens. The application of these cutting-edge technologies to existing museum collections is revealing hidden treasures of molecular information that were previously invisible to scientific investigation. The interdisciplinary collaboration required for this research brings together experts from fields as diverse as paleontology, biochemistry, material science, and molecular biology. This collaborative approach is essential for understanding the complex processes that preserve these molecular signatures and for extracting maximum information from these irreplaceable specimens. The insights gained from these collaborations are advancing our understanding of both ancient life and the fundamental processes that govern biological systems. The implications of these molecular discoveries extend far beyond the study of extinct species. The preservation mechanisms revealed by these fossils are providing insights into how biological materials can be stabilized over long time periods, with applications in medicine, biotechnology, and materials science. The understanding of ancient biochemical processes is informing research into the evolution of fundamental biological systems and the origin of life itself. The educational impact of these discoveries cannot be overstated. When students and the public learn about these preserved molecular signatures, they gain a deeper appreciation for the continuity of biochemical processes across geological time. These findings make the connection between ancient and modern life tangible and immediate, inspiring curiosity about the natural world and the processes that have shaped life on Earth. The conservation challenges associated with preserving these molecular signatures require constant attention and specialized expertise. The chemical information contained within these fossils is even more delicate than their physical structures, requiring careful control of storage conditions and handling procedures. Environmental factors that might not affect the gross anatomy of these specimens can nevertheless destroy the molecular signatures that provide the most detailed information about ancient life. The future of this research depends on continued technological advancement and interdisciplinary collaboration. As analytical techniques become more sensitive and less destructive, scientists will be able to extract ever more detailed information from these precious specimens. The development of new approaches to studying ancient biological materials promises to reveal aspects of prehistoric life that we can barely imagine today. The philosophical implications of these molecular discoveries touch on fundamental questions about the nature of biological information and its persistence through time. The preservation of chemical signatures from ancient life forms challenges our understanding of the boundaries between past and present, suggesting that the information content of life may be more persistent than we previously imagined. These fossils create direct molecular connections between ancient and modern life that transcend the usual boundaries of scientific inquiry. As we continue our quiet journey through these remarkable discoveries, we can appreciate the incredible chain of events that has preserved these molecular signatures across vast spans of geological time. The precise conditions that allowed these chemical traces to survive represent a perfect alignment of environmental factors that occurs perhaps once in millions of fossilization events. These specimens represent not just scientific curiosities, but miraculous survivals that provide unprecedented insights into the deep history of life on Earth. The study of these preserved molecular signatures reminds us that the history of life is written not just in stone, but in the very atoms and molecules that once sustained ancient creatures. These chemical traces create bridges across time that connect us directly to the biological processes that animated life millions of years ago. When we analyze these ancient molecular signatures, we are literally touching the transformed remains of the biochemical machinery that once powered movement, growth, and sensation in creatures that lived in worlds vastly different from our own. The molecular archaeology revealed by these fossils provides insights into the evolutionary continuity of life processes across geological time. The basic biochemical pathways that sustain these ancient creatures are fundamentally similar to those that sustain modern life, demonstrating the conservative nature of biological systems and the deep connections that link all living things. These molecular signatures provide direct evidence for the evolutionary relationships between ancient and modern species. The preservation of these chemical traces also provides insights into the environmental conditions that existed during the lives of these ancient animals. The isotopic signatures preserved within these molecules reflect the temperature, humidity, and atmospheric composition of the prehistoric worlds in which these creatures lived. This environmental information helps scientists reconstruct the ecological context that shaped the evolution of these remarkable species. The technological innovations required to study these molecular signatures continue to push the boundaries of analytical chemistry and material science. The development of new methods for detecting and analyzing ancient biological compounds is advancing our understanding of both preservation processes and the fundamental nature of biological systems. These technological advances have applications far beyond paleontology, contributing to progress in medicine, biotechnology, and materials engineering. The collaborative nature of this research demonstrates the power of interdisciplinary approaches to scientific inquiry. The combination of paleontological expertise, chemical analysis, and biological interpretation creates a comprehensive understanding of these ancient systems that would be impossible for any single discipline to achieve alone. This collaborative model is becoming increasingly important as scientific questions become more complex and require integration across traditional disciplinary boundaries. The discovery of these molecular signatures in fossils has also highlighted the importance of preserving and protecting fossil-bearing sites around the world. The specific conditions that created these remarkable specimens are rare and fragile, easily destroyed by human activity or natural processes. Conservation efforts must balance the need for scientific access with the protection of these irreplaceable resources for future generations. The educational value of these discoveries extends beyond formal scientific training to inspire public interest in natural history and scientific research. When people learn about these preserved molecular signatures, they gain a visceral understanding of the reality of prehistoric life that goes beyond abstract concepts. These fossils make the deep past tangible and immediate, creating connections between ancient and modern life that inspire wonder and curiosity. The museum collections that house these specimens play a crucial role in maintaining these molecular archives for future study. The proper curation and conservation of these fossils ensures that the chemical information they contain will be available for analysis using techniques that haven't been invented yet. These collections represent invaluable resources for scientific research that will continue to yield new insights for generations to come. The preparation techniques required to study these molecular signatures require exceptional skill and patience. The delicate work of extracting chemical information from ancient specimens without destroying the very molecules being studied requires careful development of new analytical protocols. The specialists who perform this work must combine expertise in chemistry, biology, and paleontology to unlock the secrets contained within these ancient treasures. The documentation and data management required for this research creates permanent records that can be shared with scientists around the world. The complex data sets generated by molecular analysis of these fossils require sophisticated database systems and analytical tools to manage and interpret. These digital resources become valuable scientific assets that can be accessed and reanalyzed as new questions arise and new techniques become available. The international collaboration required for this research brings together scientists from institutions around the globe, creating networks of expertise that transcend national boundaries. The sharing of specimens, analytical techniques, and research findings accelerates the pace of discovery and ensures that the maximum amount of information is extracted from these precious fossils. These collaborative networks represent the future of scientific research in an increasingly interconnected world. Tonight, as we rest and reflect on these extraordinary molecular discoveries, we can marvel at the remarkable preservation processes that have allowed us to study the most intimate chemical details of creatures that lived during the age of dinosaurs. These fossils represent more than scientific specimens. They are molecular time capsules that have survived impossible odds to deliver messages from the deep past about the fundamental nature of life itself. The molecular signatures preserved within these ancient specimens continue to reveal new secrets about the biochemical processes that have sustained life on Earth for billions of years. Each new analytical technique brings fresh insights into the molecular machinery that once powered these ancient creatures, creating ever more detailed pictures of how life functioned in prehistoric worlds. These discoveries remind us that the boundary between past and present is not as absolute as we might imagine, and that the molecules that once sustained ancient life continue to inform our understanding of the biological processes that sustain us today. The study of these preserved molecular signatures represents a new frontier in paleontology that promises to revolutionize our understanding of ancient life. As analytical techniques continue to improve and new approaches to studying ancient biological materials are developed, we can expect even more remarkable discoveries that will further illuminate the deep history of life on Earth. These molecular fossils create direct chemical connections between ancient and modern life that remind us of our place in the vast continuum of biological evolution. As we continue our peaceful exploration of these remarkable discoveries, we can appreciate the incredible journey that has brought these molecular treasures to our attention. From their formation in prehistoric environments to their analysis in modern laboratories, each step in their preservation and study has required perfect timing and exceptional circumstances. These specimens represent miraculous survivals from distant worlds that continue to inform our understanding of life's long history on our planet. The legacy of these molecular discoveries extends far beyond the scientific papers and museum displays they have inspired. They represent humanity's ongoing quest to understand our place in the vast sweep of natural history, connecting us to the deep molecular roots of life on Earth and reminding us that we are part of a biochemical story that began billions of years ago and continues to unfold around us today. The molecules that once sustained these ancient creatures share fundamental similarities with those that sustain us, creating tangible connections across the vast expanse of geological time. The molecular revelations hidden within these ancient specimens continue to astound researchers as they delve deeper into the chemical signatures that have somehow survived across unimaginable spans of time. When scientists examine the preserved cellular structures under the most powerful microscopes available, they discover that the fossilization process captured not just the shape of individual cells, but the actual arrangement of subcellular components that once carried out the basic functions of life. The mitochondrial signatures preserved within muscle tissues tell stories of the powerhouses that once generated energy for these ancient predators. These cellular organelles, responsible for converting nutrients into usable energy, have left behind chemical traces that reveal the metabolic intensity of these creatures. The preservation of these energy-producing structures provides direct evidence for the high-performance lifestyles that characterized these prehistoric hunters. The ribosomal signatures found within preserved tissues offer glimpses into the protein synthesis machinery that once built and maintained these ancient bodies. These molecular factories, responsible for translating genetic information into functional proteins, have left behind chemical fingerprints that revealed the sophisticated manufacturing processes that sustained life in these creatures. The detection of these protein synthesis signatures suggests that these animals had robust systems for growth, repair, and adaptation. The membrane signatures preserved within cellular structures provide insights into the barriers that once separated the internal environment of cells from the external world. These lipid-based boundaries, essential for maintaining cellular integrity and function, have left behind chemical traces that reveal the sophisticated systems these creatures used to regulate their internal chemistry. The preservation of these membrane signatures demonstrates the remarkable precision of the fossilization process. The cytoskeletal signatures found within preserved cells reveal the structural frameworks that once gave these ancient tissues their shape and mechanical properties. These protein networks, responsible for maintaining cell shape and enabling movement, have left behind chemical traces that provide insights into the biomechanical capabilities of these extinct creatures. The detection of these structural signatures helps scientists understand how these animals moved and functioned in their prehistoric environments. The nuclear signatures preserved within some cellular structures offer tantalizing glimpses into the control centers that once directed cellular activities. These command centers, containing the genetic information that guided development and function, have left behind chemical traces that may preserve echoes of the instructions that built these ancient bodies. The preservation of these nuclear signatures represents perhaps the most intimate connection we can achieve with these long-extinct creatures. The enzymatic signatures scattered throughout these preserved tissues reveal the catalytic systems that once accelerated the chemical reactions necessary for life. These biological catalysts, responsible for facilitating everything from digestion to energy production, have left behind chemical fingerprints that provide insights into the biochemical pathways that sustained these ancient animals. The detection of these enzymatic signatures demonstrates the sophisticated chemistry that powered prehistoric life. The transport protein signatures found within preserved circulatory systems reveal the molecular machinery that once moved essential substances throughout these ancient bodies. These specialized proteins, responsible for carrying oxygen, nutrients, and waste products, have left behind chemical traces that provide insights into the logistics of maintaining life in these complex organisms. The preservation of these transport signatures offers glimpses into the sophisticated distribution systems that sustained these creatures. The signaling molecule signatures preserved within neural tissues provide evidence for the communication systems that once coordinated behavior and physiological responses. These chemical messengers, responsible for transmitting information between cells and organs, have left behind traces that reveal the sophisticated coordination systems these animals used to respond to their environment. The detection of these signaling signatures provides insights into the behavioral capabilities of these extinct species. The adhesion protein signatures found within preserved tissues reveal the molecular glues that once held these ancient bodies together. These specialized proteins, responsible for maintaining tissue integrity and cellular connections, have left behind chemical traces that provide insights into the structural organization of these prehistoric creatures. The preservation of these adhesion signatures demonstrates the sophisticated architecture that supported life in these ancient animals. The regulatory protein signatures scattered throughout these specimens provide evidence for the control systems that once maintained physiological balance in these creatures. These molecular switches, responsible for turning biological processes on and off in response to changing conditions, have left behind chemical fingerprints that reveal the sophisticated homeostatic mechanisms these animals use to maintain stable internal environments. The detection of these regulatory signatures offers insights into the physiological resilience of these extinct species. The detoxification signatures, preserved within liver and kidney tissues, reveal the defensive systems that once protected these ancient animals from harmful substances. These molecular cleanup crews, responsible for neutralizing toxins and waste products, have left behind chemical traces that provide insights into the environmental challenges these creatures faced. The preservation of these detoxification signatures demonstrates the sophisticated defense mechanisms that allowed these animals to survive in complex chemical environments. The antioxidant signatures found within preserved tissues provide evidence for the protective systems that once defended these ancient cells from oxidative damage. These molecular shields, responsible for neutralizing harmful free radicals, have left behind chemical traces that reveal the sophisticated protection mechanisms these creatures use to maintain cellular health. The detection of these antioxidant signatures provides insights into the longevity and health maintenance strategies of these extinct species. The chaperone protein signatures preserved within cellular structures reveal the molecular assistance that once helped other proteins fold into their proper shapes. These biological helpers, essential for maintaining protein function and preventing cellular damage, have left behind chemical fingerprints that provide insights into the quality control systems that operated within these ancient cells. The preservation of these chaperone signatures demonstrates the sophisticated maintenance mechanisms that sustained life in these prehistoric creatures. The storage protein signatures found within preserved tissues provide evidence for the molecular warehouses that once held essential substances for future use. These biological storage systems, responsible for maintaining reserves of nutrients and other vital compounds, have left behind chemical traces that reveal the resource management strategies these creatures used to survive periods of scarcity. The detection of these storage signatures offers insights into the survival adaptations of these extinct species. The structural protein signatures preserved throughout these specimens reveal the molecular frameworks that once provided strength and flexibility to these ancient bodies. These biological building materials, responsible for creating everything from muscle fibers to bone matrix, have left behind chemical fingerprints that provide insights into the mechanical properties of these prehistoric creatures. The preservation of these structural signatures demonstrates the sophisticated engineering that supported life in these ancient animals. The sensory protein signatures found within preserved organs provide evidence for the molecular detectors that once allowed these creatures to perceive their environment. These biological sensors, responsible for detecting light, sound, chemicals, and other stimuli, have left behind chemical traces that reveal the sophisticated sensory capabilities these animals possessed. The detection of these sensory signatures provides insights into how these extinct species interacted with their prehistoric worlds. The motor protein signatures preserved within muscle tissues reveal the molecular machines that once powered movement in these ancient creatures. These biological engines, responsible for generating the forces that enabled locomotion, have left behind chemical fingerprints that provide insights into the biomechanical capabilities of these prehistoric predators. The preservation of these motor signatures demonstrates the sophisticated propulsion systems that allowed these animals to hunt and survive. The immune protein signatures scattered throughout these specimens provide evidence for the defensive systems that once protected these ancient animals from disease and infection. These biological guardians, responsible for recognizing and neutralizing threats to health, have left behind chemical traces that reveal the sophisticated immune capabilities these creatures possessed. The detection of these immune signatures offers insights into the disease-resistant strategies of these extinct species. The repair protein signatures found within preserved tissues reveal the molecular maintenance crews that once fixed damage and maintained cellular health in these ancient animals. These biological repair systems, responsible for detecting and correcting problems before they could cause serious harm, have left behind chemical fingerprints that provide insights into the self-maintenance capabilities of these prehistoric creatures. The preservation of these repair signatures demonstrates the sophisticated healing mechanisms that sustained life in these ancient species. The communication protein signatures preserved within neural and endocrine tissues provide evidence for the molecular messaging systems that once coordinated complex behaviors and physiological responses. These biological communication networks, responsible for transmitting information throughout the body, have left behind chemical traces that reveal the sophisticated coordination systems these animals used to function as integrated organisms. The detection of these communication signatures provides insights into the behavioral complexity of these extinct species. The adaptation protein signatures found within various tissues revealed the molecular flexibility systems that once allowed these ancient creatures to respond to changing environmental conditions. These biological adjustment mechanisms, responsible for modifying cellular function in response to new challenges, have left behind chemical fingerprints that provide insights into the evolutionary adaptability of these prehistoric animals. The preservation of these adaptation signatures demonstrates the sophisticated response systems that enabled these species to survive in dynamic environments. The timing protein signatures preserved within cellular structures provide evidence for the molecular clocks that once coordinated the daily rhythms and seasonal cycles of these ancient creatures. These biological timekeepers, responsible for maintaining proper timing of physiological processes, have left behind chemical traces that reveal the sophisticated temporal organization systems these animals possessed. The detection of these timing signatures offers insights into the behavioral rhythms of these extinct species. The growth factor signatures found within preserved tissues reveal the molecular signals that once guided the development and growth of these ancient animals. These biological growth regulators, responsible for controlling cell division and tissue formation, have left behind chemical fingerprints that provide insights into the developmental processes that shaped these prehistoric creatures. The preservation of these growth factor signatures demonstrates the sophisticated development programs that created these complex organisms. The stress response signatures preserved throughout these specimens provide evidence for the molecular emergency systems that once helped these ancient animals cope with dangerous situations. These biological crisis management systems, responsible for mobilizing resources and adjusting physiology during times of threat, have left behind chemical traces that reveal the sophisticated survival mechanisms these creatures possessed. The detection of these stress response signatures provides insights into the challenges these extinct species faced in their daily lives. The aging signatures found within preserved tissues reveal the molecular processes that once controlled the lifespan and senescence of these ancient creatures. These biological aging mechanisms, responsible for the gradual changes that occurred over the lifetime of these animals, have left behind chemical fingerprints that provide insights into the longevity strategies of these prehistoric species. The preservation of these aging signatures demonstrates the sophisticated life history programs that governed the existence of these ancient animals. The reproductive signals preserved within specialized tissues provide evidence for the molecular systems that once controlled breeding behavior and reproductive success in these ancient creatures. These biological reproductive controllers responsible for coordinating mating behaviors and reproductive physiology have left behind chemical traces that reveal the sophisticated breeding strategies these animals employed. The detection of these reproductive signatures offers insights into the evolutionary success mechanisms of these extinct species. The territorial marking signatures found within preserved scent glands reveal the molecular communication systems that once allowed these ancient animals to establish and maintain territories. These biological marking systems, responsible for chemical communication about ownership and boundaries, have left behind chemical fingerprints that provide insights into the social organization of these prehistoric creatures. The preservation of these territorial signatures demonstrates the sophisticated social systems that structured ancient communities. The parental care signatures preserved within certain specimens provide evidence for the molecular systems that once guided nurturing behaviors in these ancient animals. These biological parenting controllers, responsible for coordinating caregiving behaviors and protective responses, have left behind chemical traces that reveal the sophisticated family structures these creatures may have maintained. The detection of these parental care signatures provides insights into the social complexity of these extinct species. The learning and memory signatures found within preserved brain tissues reveal the molecular systems that once allowed these ancient creatures to acquire and retain information about their environment. These biological learning mechanisms, responsible for adapting behavior based on experience, have left behind chemical fingerprints that provide insights into the cognitive capabilities of these prehistoric animals. The preservation of these learning signatures demonstrates the sophisticated information processing systems that enabled these species to survive and thrive. The problem-solving signatures preserved within neural structures provide evidence for the molecular systems that once enabled these ancient animals to navigate complex challenges in their environment. These biological problem-solving mechanisms, responsible for generating creative solutions to survival challenges, have left behind chemical traces that reveal the sophisticated cognitive abilities these creatures possessed. The detection of these problem-solving signatures offers insights into the intelligence levels of these extinct species. The environmental sensing signatures found throughout these preserved specimens reveal the molecular systems that once allowed these ancient animals to monitor and respond to changing conditions in their world. These biological environmental monitors, responsible for detecting changes in temperature, humidity, chemistry, and other factors, have left behind chemical fingerprints that provide insights into the environmental awareness of these prehistoric creatures. The preservation of these sensing signatures demonstrates the sophisticated environmental monitoring systems that helped these species survive. The energy conservation signatures preserved within various tissues provide evidence for the molecular systems that once helped these ancient animals manage their energy resources efficiently. These biological energy management systems, responsible for optimizing energy use and storage, have left behind chemical traces that reveal the sophisticated metabolic strategies these creatures employed. The detection of these energy conservation signatures provides insights into the survival economics of these extinct species. The wound healing signatures found within preserved tissues reveal the molecular systems that once repaired injuries and maintained tissue integrity in these ancient animals. These biological healing mechanisms, responsible for restoring damaged structures and preventing infection, have left behind chemical fingerprints that provide insights into the recovery capabilities of these prehistoric creatures. The preservation of these healing signatures demonstrates the sophisticated repair systems that maintained health in these ancient species. The toxin-resistant signatures preserved within detoxification organs provide evidence for the molecular systems that once protected these ancient animals from environmental poisons and harmful substances. These biological protection mechanisms, responsible for neutralizing dangerous compounds, have left behind chemical traces that reveal the sophisticated defense strategies these creatures employed against chemical threats. The detection of these toxin-resistant signatures offers insights into the environmental challenges these extinct species faced. The pathogen-resistant signatures found within immune tissues reveal the molecular systems that once protected these ancient animals from infectious diseases and parasites. These biological defense mechanisms, responsible for recognizing and eliminating harmful microorganisms, have left behind chemical fingerprints that provide insights into the disease resistance capabilities of these prehistoric creatures. The preservation of these pathogen-resistant signatures demonstrates the sophisticated immune systems that protected these ancient species. The temperature regulation signatures preserved throughout these specimens provide evidence for the molecular systems that once helped these ancient animals maintain optimal body temperatures. These biological thermoregulation mechanisms, responsible for adjusting heat production and heat loss, have left behind chemical traces that reveal the sophisticated temperature control strategies these creatures employed. The detection of these temperature regulation signatures provides insights into the thermal physiology of these extinct species. The water balance signatures found within preserved organs reveal the molecular systems that once maintained proper hydration and fluid balance in these ancient animals. These biological water management systems, responsible for regulating water intake, retention, and excretion, have left behind chemical fingerprints that provide insights into the osmoregulatory capabilities of these prehistoric creatures. The preservation of these water balance signatures demonstrate the sophisticated fluid control systems that sustained life in these ancient species. The pH regulation signatures preserved within cellular structures provide evidence for the molecular systems that once maintained proper acid-base balance in these ancient animals. These biological pH control mechanisms, responsible for buffering against harmful changes in acidity, have left behind chemical traces that reveal the sophisticated chemical balance strategies these creatures employed. The detection of these pH regulation signatures offers insights into the biochemical stability systems of these extinct species. The mineral transport signatures found throughout these preserved specimens reveal the molecular systems that once delivered essential elements to the tissues of these ancient animals. These biological mineral distribution systems, responsible for transporting calcium, iron, phosphorus, and other vital elements, have left behind chemical fingerprints that provide insights into the nutritional physiology of these prehistoric creatures. The preservation of these mineral transport signatures demonstrates the sophisticated nutrient delivery systems that sustained these ancient species. The waste elimination signatures preserved within excretory organs provide evidence for the molecular systems that once removed harmful waste products from the bodies of these ancient animals. These biological waste management systems, responsible for filtering and eliminating toxic substances, have left behind chemical traces that reveal the sophisticated detoxification strategies these creatures employed. The detection of these waste elimination signatures provides insights into the metabolic waste processing capabilities of these extinct species. The tissue maintenance signatures found within preserved structures reveal the molecular systems that once kept these ancient animals' bodies in good repair and working order. These biological maintenance mechanisms, responsible for ongoing cellular renewal and tissue replacement, have left behind chemical fingerprints that provide insights into the longevity strategies of these prehistoric creatures. The preservation of these tissue maintenance signatures demonstrates the sophisticated self-repair systems that sustained life in these ancient species. The developmental control signatures, preserved within growth zones, provide evidence for the molecular systems that once guided the transformation of these ancient animals from juveniles to adults. These biological development controllers, responsible for coordinating growth patterns and maturation processes, have left behind chemical traces that reveal the sophisticated developmental programs these creatures possessed. The detection of these developmental control signatures offers insights into the life history strategies of these extinct species. The behavioral control signatures found within neural tissues reveal the molecular systems that once coordinated the complex behaviors that allowed these ancient animals to survive and thrive. These biological behavior controllers, responsible for generating appropriate responses to environmental stimuli, have left behind chemical fingerprints that provide insights into the behavioral repertoires of these prehistoric creatures. The preservation of these behavioral control signatures demonstrates the sophisticated action control systems that enabled these species to succeed in their environments. The social coordination signatures preserved within communication organs provide evidence for the molecular systems that once facilitated group behaviors and social interactions in these ancient animals. These biological social controllers, responsible for coordinating activities with other individuals, have left behind chemical traces that reveal the sophisticated social systems these creatures may have maintained. The detection of these social coordination signatures provides insights into the community structures of these extinct species. The predator detection signatures found within sensory organs revealed the molecular systems that once allowed these ancient animals to identify and respond to threats in their environment. These biological threat detection systems, responsible for recognizing danger and triggering appropriate responses, have left behind chemical fingerprints that provide insights into the survival strategies of these prehistoric creatures. The preservation of these predator detection signatures demonstrates the sophisticated danger awareness systems that protected these ancient species. The prey detection signatures preserved within hunting-related tissues provide evidence for the molecular systems that once helped these ancient predators locate and capture their food. These biological hunting systems, responsible for detecting and tracking prey animals, have left behind chemical traces that reveal the sophisticated predation strategies these creatures employed. The detection of these prey detection signatures offers insights into the hunting capabilities of these extinct species. The navigation signatures found within orientation organs reveal the molecular systems that once allowed these ancient animals to find their way through complex environments. These biological navigation systems, responsible for spatial orientation and directional movement, have left behind chemical fingerprints that provide insights into the mobility strategies of these prehistoric creatures. The preservation of these navigation signatures demonstrates the sophisticated guidance systems that enabled these species to move efficiently through their world. The memory consolidation signatures preserved within brain tissues provide evidence for the molecular systems that once allowed these ancient animals to store and retrieve important information about their environment. These biological memory systems, responsible for learning from experience and applying knowledge to new situations, have left behind chemical traces that reveal the sophisticated information processing capabilities these creatures possessed. The detection of these memory consolidation signatures provides insights into the cognitive abilities of these extinct species. The decision-making signatures found within neural structures reveal the molecular systems that once enabled these ancient animals to choose appropriate responses to complex situations. These biological decision systems, responsible for weighing options and selecting optimal behaviors, have left behind chemical fingerprints that provide insights into the cognitive sophistication of these prehistoric creatures. The preservation of these decision-making signatures demonstrates the sophisticated choice-making systems that guided the behavior of these ancient species. The pattern recognition signatures preserved within sensory processing areas provide evidence for the molecular systems that once allowed these ancient animals to identify important features in their environment. These biological pattern detection systems, responsible for recognizing significant shapes, sounds, and other stimuli, have left behind chemical traces that reveal the sophisticated perception capabilities these creatures possessed. The detection of these pattern recognition signatures offers insights into the sensory processing abilities of these extinct species. The attention control signatures found within brain tissues reveal the molecular systems that once allowed these ancient animals to focus their mental resources on important aspects of their environment. These biological attention systems, responsible for directing cognitive resources toward relevant stimuli, have left behind chemical fingerprints that provide insights into the mental focus capabilities of these prehistoric creatures. The preservation of these attention control signatures demonstrates the sophisticated cognitive control systems that enabled these species to process information efficiently. The emotional regulation signatures preserved within limbic structures provide evidence for the molecular systems that once controlled the emotional responses of these ancient animals. These biological emotion controllers, responsible for generating appropriate emotional reactions to different situations, have left behind chemical traces that revealed the sophisticated emotional systems these creatures possessed. The detection of these emotional regulation signatures provides insights into the psychological complexity of these extinct species. The motivation signatures found within reward processing areas reveal the molecular systems that once drove the goal-directed behaviors of these ancient animals. These biological motivation systems, responsible for generating the drive to pursue important objectives, have left behind chemical fingerprints that provide insights into the behavioral motivation strategies of these prehistoric creatures. The preservation of these motivation signatures demonstrates the sophisticated drive systems that powered the activities of these ancient species. The coordination signatures preserved within motor control areas provide evidence for the molecular systems that once enabled these ancient animals to execute complex, precise movements. These biological coordination systems, responsible for integrating sensory information with motor output, have left behind chemical traces that reveal the sophisticated movement control capabilities these creatures possessed. The detection of these coordination signatures offers insights into the motor skills of these extinct species. The balance signatures found within vestibular structures reveal the molecular systems that once maintained postural stability and spatial orientation in these ancient animals. These biological balance systems, responsible for detecting body position and movement, have left behind chemical fingerprints that provide insights into the equilibrium maintenance strategies of these prehistoric creatures. The preservation of these balance signatures demonstrates the sophisticated stability control systems that enabled these species to move gracefully through their environment. The reflexive response signatures preserved within spinal cord tissues provide evidence for the molecular systems that once generated rapid, automatic responses to stimuli in these ancient animals. These biological reflex systems, responsible for producing immediate reactions to important stimuli, have left behind chemical traces that reveal the sophisticated automatic response capabilities these creatures possessed. The detection of these reflexive response signatures provides insights into the survival reaction systems of these extinct species. The integration signatures found throughout neural networks reveal the molecular systems that once coordinated the complex interactions between different brain regions in these ancient animals. These biological integration systems, responsible for combining information from multiple sources, have left behind chemical fingerprints that provide insights into the neural coordination capabilities of these prehistoric creatures. The preservation of these integration signatures demonstrates the sophisticated neural networking systems that enabled these species to function as coordinated organisms. The plasticity signatures preserved within neural tissues provide evidence for the molecular systems that once allowed these ancient animals to adapt their neural connections in response to experience. These biological plasticity systems, responsible for modifying neural circuits based on learning and development, have left behind chemical traces that reveal the sophisticated neural adaptation capabilities these creatures possessed. The detection of these plasticity signatures offers insights into the learning and development potential of these extinct species. The efficiency signatures found within metabolic pathways reveal the molecular systems that once optimized energy use and resource allocation in these ancient animals. These biological efficiency systems, responsible for maximizing the benefit obtained from available resources, have left behind chemical fingerprints that provide insights into the metabolic optimization strategies of these prehistoric creatures. The preservation of these efficiency signatures demonstrates the sophisticated resource management systems that enabled these species to thrive in competitive environments. The resilient signatures preserved throughout these specimens provide evidence for the molecular systems that once helped these ancient animals recover from stress, injury, and other challenges. These biological resilient systems, responsible for maintaining function despite adversity, have left behind chemical traces that reveal the sophisticated recovery capabilities these creatures possessed. The detection of these resilient signatures provides insights into the survival strength of these extinct species. The adaptation signatures found within regulatory systems reveal the molecular mechanisms that once allowed these ancient animals to adjust their physiology and behavior in response to changing environmental conditions. These biological adaptation systems, responsible for modifying function to match new circumstances, have left behind chemical fingerprints that provide insights into the evolutionary flexibility of these prehistoric creatures. The preservation of these adaptation signatures demonstrates the sophisticated adjustment capabilities that enabled these species to survive in dynamic environments. As we continue our peaceful journey through these molecular discoveries, we can appreciate how each preserved chemical signature adds another layer to our understanding of these ancient creatures. The combination of all these molecular traces creates a comprehensive picture of prehistoric life that extends far beyond what bones alone could ever reveal. These chemical fossils provide direct evidence for the sophisticated biological systems that once powered movement, sensation, and thought in animals that lived when our world was young and vastly different. The preservation of these molecular signatures across such vast spans of time represents one of the most remarkable achievements in the history of fossilization. The precise conditions that allowed these delicate chemical traces to survive the passage of millions of years required a perfect alignment of environmental factors that occurs perhaps once in countless millions of preservation events. Each specimen represents not just a scientific treasure, but a miraculous survivor that has beaten astronomical odds to deliver its molecular message from the deep past. The analytical techniques required to detect and study these ancient molecular signatures continue to advance at an extraordinary pace. Each new technological development brings fresh capabilities for extracting information from these precious specimens, revealing previously hidden aspects of ancient biochemistry. The application of these cutting-edge methods to museum collections around the world is uncovering molecular treasures that have been waiting patiently for the right technology to reveal their secrets. The collaborative nature of this research brings together expertise from numerous scientific disciplines, creating research teams that combine paleontological knowledge with advanced chemistry, molecular biology, and materials science. This interdisciplinary approach is essential for understanding the complex processes that preserve these molecular signatures and for extracting maximum information from these irreplaceable specimens. The insights gained from these collaborations are advancing our understanding of both ancient life and the fundamental processes that govern biological systems. The implications of these molecular discoveries extend far beyond the study of extinct species. The preservation mechanisms revealed by these fossils are providing insights into how biological materials can be stabilized over geological time periods, with applications in medicine, biotechnology, and materials science. The understanding of ancient biochemical processes is informing research into the evolution of fundamental biological systems and the origin of life itself. The educational impact of these discoveries cannot be overstated when students and the public learn about these preserved molecular signatures. They gain a visceral understanding of the continuity of life processes across geological time. These findings make the connection between ancient and modern life tangible and immediate, inspiring curiosity about the natural world and the processes that have shaped life on Earth. The conservation challenges associated with preserving these molecular signatures require constant attention and specialized expertise. The chemical information contained within these fossils is even more delicate than their physical structures, requiring careful control of storage conditions and handling procedures. Environmental factors that might not affect the gross anatomy of these specimens can nevertheless destroy the molecular signatures that provide the most detailed information about ancient life. The future of this research depends on continued technological advancement and interdisciplinary collaboration. As analytical techniques become more sensitive and less destructive, scientists will be able to extract ever more detailed information from these precious specimens. The development of new approaches to studying ancient biological materials promises to reveal aspects of prehistoric life that we can barely imagine today. The philosophical implications of these molecular discoveries touch on fundamental questions about the nature of biological information and its persistence through time. The preservation of chemical signatures from ancient life forms challenges our understanding of the boundaries between past and present, suggesting that the information content of life may be more persistent than we previously imagined. These fossils create direct molecular connections between ancient and modern life that transcend the usual boundaries of scientific inquiry. Tonight, as we rest and reflect on these extraordinary molecular discoveries, we can marvel at the remarkable preservation processes that have allowed us to study the most intimate chemical details of creatures that lived during the age of dinosaurs. These fossils represent more than scientific specimens. They are molecular time capsules that have survived impossible odds to deliver messages from the deep past about the fundamental nature of life itself. The molecular signatures preserved within these ancient specimens continue to reveal new secrets about the biochemical processes that sustained life millions of years ago. Each new analytical technique brings fresh insights into the molecular machinery that once powered these ancient creatures, creating ever more detailed pictures of how life functioned in prehistoric worlds. These discoveries remind us that the boundary between past and present is not as absolute as we might imagine, and that the molecules that once sustained ancient life continue to inform our understanding of the biological processes that sustain us today. The study of these preserved molecular signatures represents a new frontier in paleontology that promises to revolutionize our understanding of ancient life. As analytical techniques continue to improve and new approaches to studying ancient biological materials are developed, we can expect even more remarkable discoveries that will further illuminate the deep history of life on Earth. These molecular fossils create direct chemical connections between ancient and modern life that remind us of our place in the vast continuum of biological evolution. The quantum-level preservation found within some of these specimens reveals molecular arrangements that have remained stable across geological timescales that dwarf human comprehension. When scientists examine these fossils using the most advanced spectroscopic techniques available, they discover that the replacement minerals formed in patterns that mirror the original quantum mechanical properties of biological molecules. This level of preservation suggests that information can persist at the most fundamental levels of matter organization. The crystallographic signatures preserved within these mineralized tissues tell stories of the ordered arrangements that once characterized living systems. The specific orientations of calcium phosphate crystals within fossilized bone reflect the original collagen fiber arrangements that gave these structures their strength and flexibility. These crystalline patterns provide insights into the biomechanical properties of ancient tissues that would be impossible to determine from gross anatomy alone. The electromagnetic signatures detected within some specimens provide evidence for the electrical properties that once characterized living neural tissue. The arrangement of iron-rich minerals within fossilized brain structures creates patterns that suggest these tissues once conducted electrical impulses, similar to those found in modern nervous systems. These electromagnetic traces provide direct evidence for the bioelectrical activity that once animated these ancient minds. The thermodynamic signatures preserved within metabolic tissues revealed the energy landscapes that once drove biochemical processes in these ancient creatures. The specific arrangements of phosphate minerals within fossilized muscle cells reflect the original energy gradients that powered contraction and movement. These thermodynamic patterns provide insights into the metabolic efficiency and power output capabilities of these extinct species. The information theory signatures found within preserved genetic material structures suggest that biological information storage and processing systems in these ancient creatures were far more sophisticated than previously imagined. The statistical patterns observed in mineralized cellular components indicate that these organisms possessed complex information processing capabilities that approached those found in modern biological systems. The network topology signatures preserved within neural structures reveal the connectivity patterns that once characterized the brains of these ancient animals. The branching patterns of mineralized nerve fibers create mathematical networks that can be analyzed using modern graph theory techniques. These network analyses provide insights into the computational capabilities and information processing architectures of these extinct species. The fractal signatures found throughout these preserved biological structures demonstrate that these ancient organisms exhibited the same types of self-similar patterns that characterize modern living systems. The branching patterns of blood vessels, the arrangements of muscle fibers, and the organization of neural networks all display fractal geometries that suggest these creatures had evolved sophisticated strategies for maximizing surface area and efficiency within limited volumes. The chaos theory signatures detected within preserved physiological systems provide evidence for the complex dynamical behaviors that once characterized these ancient organisms. The irregular patterns observed in mineralized heart muscle suggest that these creatures had cardiovascular systems capable of exhibiting the same types of complex rhythms found in modern animals. These chaotic signatures provide insights into the dynamical stability and adaptability of ancient physiological systems. The quantum coherence signatures preserved within some neural structures suggest that these ancient brains may have utilized quantum mechanical effects for information processing. The specific arrangements of minerals within fossilized neural tissue create patterns that are consistent with quantum coherence effects that might have enhanced computational capabilities. These quantum signatures provide tantalizing hints about the fundamental nature of consciousness and cognition in these extinct species. The holographic signatures found within preserved memory structures suggest that these ancient animals may have stored information in distributed patterns throughout their neural networks. The interference patterns observed in mineralized brain tissue are consistent with holographic information storage systems that would have been highly resistant to damage and degradation. These holographic signatures provide insights into the information storage strategies of these extinct species. The emergent property signatures detected throughout these preserved biological systems demonstrate that these ancient organisms exhibited complex behaviors that arose from the interactions of simpler components. The patterns observed in mineralized tissue arrangements suggest that these creatures had evolved sophisticated strategies for generating complex behaviors from relatively simple neural and muscular elements. The self-organization signatures preserved within developmental structures reveal the autonomous assembly processes that once guided the growth and development of these ancient creatures. The patterns observed in mineralized growth zones suggest that these organisms had evolved sophisticated self-organizing systems that could generate complex structures without external guidance or control. The adaptive complexity signatures found throughout these specimens demonstrate that these ancient organisms had evolved sophisticated strategies for maintaining function despite environmental challenges and internal perturbations. The patterns observed in preserved physiological systems suggest that these creatures possessed robust adaptive mechanisms that could maintain performance across a wide range of conditions. Unlock Earth's ancient secrets. Dive deeper into the wonders of prehistoric life preserved in stone. Subscribe for more fascinating paleontological discoveries, exclusive content, and expert insights. Don't miss out. Join our journey through time today. Explore now and connect with a world millions of years in the making.